Hi all, thank you so much for joining us today. My name is Victoria Gem, and I'm an Assistant Research Director here at the Duke-Margolis Institute for Health Policy. Welcome to our workshop on opportunities to improve dose finding and optimization for rare disease drug development, which is being convened by Duke-Margolis and our partners at the U.S. Food and Drug Administration. During this virtual workshop, we'll have 5 sessions with presentations and moderated discussion, spanning a range of topics related to dose finding and optimization for rare disease drug development. We'll hear from an array of experts to better understand the burdens faced by patients and caregivers when participating in rare disease clinical trials, the challenges and considerations associated with dose finding and optimization for rare disease populations, and opportunities to enhance dose finding practices in such trials. Before we begin, I'd like to quickly go over some housekeeping notes. We'd like to take a moment to acknowledge our commitment to academic independence and to avoid taking partisan positions in our work. You can find more information about our policies on our website listed on this page, and where you also found information for the event today. Here are a few reminders about our meeting format that will help us organize discussion in the virtual setting. So as you hopefully saw, the materials for today's event are available on the Duke-Margolis event webpage. To our attendees today, if you would like to ask a question during the presentations and open discussion portion of the meeting, please type it into the Zoom Q&A box. We really look forward to your active participation via questions, and we'll incorporate as many as possible during today's discussions. We do certainly have a packed agenda, and due to the high volume of meeting participants, we may not be able to get to your question. A recording of the meeting will be available on the Duke-Margolis event webpage and YouTube channel after the conclusion of the event. And then finally, the Duke-Margolis Institute is convening today's meeting in collaboration with the FDA. This is not a federal advisory committee meeting. While we are keenly interested in your input on this topic, we won't be following advisory committee procedures or taking a vote at the end of the meeting. This meeting will instead be a success if there is an exchange of a lot of ideas and open discussion. We are thrilled that you all are able to join us today, and we anticipate an interesting and productive discussion. One last thing I want to cover before we jump into the meeting is today's agenda. We're going to begin today's workshop with a brief high-level presentation from the FDA on some of the major challenges with dose finding and optimization for therapies intended for rare diseases. We'll then move on to a panel discussion between patients and caregivers to hear their lived experiences participating in rare disease clinical trials. Panelists will discuss the burdens commonly faced by patients and their families, as well as opportunities to reduce barriers to participation in dose-finding trials. Next, we'll bring together a group of experts to discuss novel statistical methods and trial design approaches to support dose optimization studies, as well as the role of non-clinical models in informing early phase trial design. After a quick break, we'll hear from a group of experts on some of the considerations for selecting clinical outcome assessments and sensitive endpoints for small and complex populations. We'll then explore opportunities for use of real-world data to enhance and inform optimization in the post-market setting. And after another quick break, we'll end our workshop with a case study presentation and discussion on next steps that will aim to tie together the topics that arise throughout the workshop today. We'll then adjourn at 3:30 p.m. Eastern Time. It's quite a packed agenda, but we're really looking forward to such a wonderful, productive conversation today. And now I'd like to invite Dr. Isam Zanaya, Director of Office of Clinical Pharmacology at the FDA, to kick us off with the introductory presentation. I'll turn it over to you, Isam.
Thanks, Victoria. And if I could have the next slide, please. Great. Thank you to the colleagues at Duke-Margolis and the FDA organizers for inviting me to provide some high-level contextual remarks over the next 10 minutes or so. If I could have the next slide, please. So I want to start by stating what I think is probably obvious, if not intuitive, to everybody that's tuning in today, and that is, at the heart of it, drug development and regulatory evaluation of new therapeutics, for that matter, comes down to 2 key questions. On the left-hand side is the question of access. When one looks at the totality of the safety, efficacy, and product quality data does the investigational new drug demonstrate a favorable risk-benefit profile such that the appropriate public health decision would be to make that treatment available to patients. That is through the approval of the marketing application. On the right-hand side is a question of therapeutic optimization. Have we sufficiently elucidated the determinants of treatment response variability in a way that allows us to optimize therapeutic use at the individual patient level? And in an ideal situation, these equally important questions are answerable, robustly answerable during drug development. However, there can be a tension between these questions of access and optimization. And often when we're talking about optimization, we're talking about dose optimization. Next slide, please. I am only leaving this up here to refer to it. If you want to hear a little bit more about precision dosing or read a little bit more about this, there's a paper that we published a few years ago, but in the interest of time, I want to skip this and go to the next slide, which talks a little bit more about that tension that I mentioned. When might this tension occur? In the development of new therapeutics for common chronic diseases, we have the opportunity to conduct clinical pharmacology studies. They're routinely performed in healthy volunteers, in patients, in important subpopulations to evaluate variability, usually pharmacokinetic variability within and across various samples of the population. This is what's shown on the left hand of the slide. And when this is coupled with our understanding of the relationship between a drug's exposure, drug concentration, and response, whether that's efficacy response or safety, we are then able to derive dosing recommendations for these patient populations. Once a drug is approved and used in even more diverse patient populations, this is in the second panel, we may be able to enhance our understanding of response variability, expand our understanding of dose exposure response relationships, and bring a more precise approach to dosing over the more classical kind of empirical trial and error approach that's very common in the clinic. And this is what you see on the right-hand side. And even in this scenario of common chronic diseases, which is often characterized by good sample sizes prior to drug approval and a relatively large denominator of patients getting treated after the drug is approved, we're often faced, at least in the post-approval setting, with this challenge of limited integrative data sets that include well-curated patient information, drug use data, biospecimens, and informatics capabilities. And maybe we'll talk a little bit today about how those can be improved upon, leveraged for the rare disease context. Next slide, please. So that was for common chronic diseases. Is rare diseases exceptional? And we can say that in this situation, we may not, and we often don't have the numbers to unequivocally answer questions about dosing and regimen optimization. And we have this tremendously urgent specter of unmet medical need to contend with. In fact, even the question of determining drug efficacy is often a challenge in the rare disease setting. So just to orient you from left to right here, we go from conventional drug development approaches for high prevalence indications to ever smaller populations, with the major differences, not getting into too much detail, going across these spaces, often being the size of the so-called phase 2 or dose ranging study, if there even is one, more accurately, the nature of the dose finding exercise, differences in the basis for the evidence of effectiveness determination, and the nature of the non-clinical toxicology studies. And the further right you travel here, as you truly get to N of 1 therapeutics, if you can click, please, for me. N of 1 therapeutics, where we're talking about, imminently fatal conditions for patients that, one or two patients that have molecular aberration or genetic lesion. There's even more tension when we're talking about the sort of access side of the equation and the dose therapeutic optimization side. In fact, in these scenarios, there's a whole slew of challenges related to patients and caregivers being even able to access systems of care, along with a range of economic, regulatory, and humanistic considerations that often overwhelm questions of dose optimization that we laid out in this table and in this publication earlier in the year. Next slide, please. So there is a kind of an exceptionalism in rare disease. That said, I think we have to be very skeptical of any position that deprioritizes good dose finding in the rare disease drug development context and dose optimization for patients because of some claim that patients care more about access, including sometimes to unproven therapies or marginally effective ones, that they care more about access than they do about getting the dose right. In fact, we have taken that paternalistic posture before, and engagement with patients with severe and life-threatening conditions doesn't necessarily reveal, that preference to be the case. For example, when we look at HIV, in the early days of HIV and AIDS, patients were very clear that optimal regimens, including optimal dosing, was of critical importance to them. Even in oncology, where it's long been presumed that patients would desire a drug with any survival advantage, we see a very clear patient voice that says they're not willing to trade quality of life necessarily for quantity, that it's not acceptable to many patients to take a new drug that may nominally prolong life if that survival benefit is marked by, for example, toxicities that prevent them from doing the things they want to do with the time they've gained. In fact, when you look at the newest HIV guidelines, they cite many patient-centered reasons to consider regimen optimization, and many of these reasons, which are in the middle of the slide, are relevant for dose optimization. They're relevant for many diseases, including rare diseases. And while, of course, clinical benefit is a priority, patients also value regimens that reduce pill burden or reduce dosing frequency, regimens that enhance tolerability and or decrease either the frequency or the severity of short and long-term toxicities, that prevent or mitigate drug-drug interactions, that would eliminate food requirements, that could allow for optimal use during pregnancy or when pregnancy is desired or may occur, and of course that reduce costs. So, some of these concerns are addressable through dose optimization. Next slide, please. So exceptional, yes. Unprecedented, no. And insurmountable, no. The challenges in rare disease drug development notwithstanding, we've shown that good dose finding is possible in the rare disease settings. These are just a sampling of papers that our staff in the Office of Clinical Pharmacology, along with other collaborators, have published that describe pragmatic approaches for dose selection and dose optimization in rare disease. And one hope for today is to discuss how we might bring to bear all the tools we have at our disposal to make this process more efficient in this high stakes enterprise. Next slide, please. And this is my final slide. It highlights what might be the opportunities and the considerations. From an evidentiary standpoint, we're often faced with three issues in rare disease drug development and regulatory evaluation. And that is the biomarker or the clinical outcome assessment or the clinical endpoint fit for its intended purpose in that drug development context? What constitutes confirmatory evidence of effectiveness? And of course, is the dose adequate and optimal for patients? And in many ways, the same approaches can be leveraged to address all of these questions. And they include optimized clinical trial designs, biomarker discovery and validation, the development and application of non-clinical models of disease, as well as drug response, and quantitative methods, including empirical and mechanistic modeling and simulation strategies, as well as the use of real-world data. Importantly, I've drawn these sort of interconnected lines. These shouldn't be considered in isolation insofar as no one approach might be sufficient in terms of information on its own, and if approached holistically, each can more impactfully inform the other. So today we look forward to hearing from thought leaders in many of these areas with hopes that the discussion will offer useful paths to patient-centered dose finding and optimization in rare disease. Looking forward to the rest of the day, and thank you all for being here.
Wonderful. Thank you so much, Isam, for providing a really wonderful level setting presentation that we can continue to build on throughout this meeting today. So now we're going to transition into our first session of the day, which is titled Patient and Clinical Perspectives on Dose Finding and Optimization in Rare Disease Drug Development. I have the honor of moderating this panel session, and I'd like to invite all our panelists for this first portion of the day to come onto screen at this time. I'm thrilled to be moderating where we'll be joined by a number of excellent panelists. So before we move on to moderated discussion, I want to make sure all of our speakers for this session have the opportunity to share some brief remarks. So I'd like to first introduce Dr. Daniel Boyce, Education Director of the Biomedical and Health Data Sciences Collaborative at Tufts University School of Medicine. Danielle, whenever you're ready, please feel free to give your opening remarks.
Thank you very much. And for the purposes of this discussion, I'm just Charlie's mom, not Dr. Boyce. And do want to acknowledge that privilege. So when I talk about barriers, my barriers are not health insurance, are not lack of education or access or connections. In fact, I even work in neurology research. So Charlie had a devastating form of epilepsy called infantile spasms when he was a baby, and has had a better part of the right side of his brain removed in the hopes of slowing down these seizures. And Charlie still has a lot of seizures and is at high risk of dying from a seizure. So I'm interested in drugs that would stop those seizures that he's having. And in fact, every morning for just about the last 14 years, I've held my breath until I could confirm that Charlie was still alive and didn't die in his sleep. So the urgency is real for me. And a few months ago, I was thrilled to hear that he might be eligible for a clinical trial because we don't have a lot of clinical trials in Charlie's world. But then my heart sank when I found out they needed an EEG for the screening, because an EEG is a test that for someone like Charlie, who has more than just seizures. These diseases are more than just seizures, but their behaviors and cognitive impairment, intellectual disability, autism, agitation, mobility challenges. This is a big lift. And it's not because I couldn't take the day off from work or pay for the travel or his aid or anything like that. It was literally getting that reading, that clinical reading, and took about a week of back and forth with the child life department at our wonderful children's hospital to make a plan. But it could have just as easily not happened, and we might not have gotten in through the screening as a result. And so, I want to talk about the other issues that, as part of this ecosystem of his disease, I call it. It's not just these seizures, but it's all the devastating things that those seizures caused. And for families like mine, when you're talking about dose, we don't have a lot of wiggle room, even on benign side effects, seemingly benign. So you might think, oh, make word retrieval difficult, can't remember your words. Well, for a child who only has a couple words, that's devastating. Is that worth it? I don't know. Right? Is that the right dose? Maybe not for us. And so understanding that if he's extra sleepy, that might mean he can't go to school. And he needs that, right? So these are the kinds of things that I encourage you to understand about our families, that it's not just this one symptom, but often these diseases are so noble and heroic to study because of how devastating they are to the entire family. And finally, Wendy Ross coined a phrase that I love: invisible ramps. If I'm going someplace with Charlie and he needs his wheelchair that day, and I asked, do you have a ramp? Is it accessible? Of course, right? Of course, that's something that you will accommodate. You understand that that's important, but Charlie needs invisible ramps. in all of these studies, in his clinical care, and in his whole life. There are lots of things that he needs that are invisible maybe to you, that these accommodations, and it's part of working with the community and the family to truly understand, so that you can have a successful study, because it's not because the families don't want to participate, but sometimes the actual reading or the measurement is impossible to obtain. And finally, I do also want to mention that when you have a child with communication barriers, or an adult with communication challenges, some of the instruments are only validated for patients and not caregivers. And so if you want to know what Charlie thinks, you have to go through me, unfortunately. And so just keeping that in mind, again, understanding the full disease before you study it. Thank you.
Great. Thank you so much, Danielle. We really appreciate you sharing your personal experience and look forward to hearing more soon. So next, I'd like to welcome Dr. Allison Brent, Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics. Allison, whenever you're ready, please feel free to give your remarks.
Great. Thank you so much for the invitation, for being here, and hopefully I can share a perspective not unlike Danielle's, but also in many ways quite different. So as mentioned, I am the Chief Science Officer at the Foundation for Angelman Syndrome Therapeutics, or FAST. But I started my career and I continue my career as a veterinarian. So I'm a veterinarian who's been working in medical device development, running clinical trials on animals with naturally occurring diseases where there's a huge clinical unmet need, in order to advance those devices to the pediatric population, where there's a very significant clinical unmet need. And that's where my whole career was until 2014, when my daughter was diagnosed with Angelman syndrome, which is a random monogenic neurodevelopmental disorder that's not degenerative, but incredibly and severely debilitating. And when she was diagnosed with Angelman syndrome, what I did as a scientist and clinician, probably similar to Danielle and Justin and many people on this panel, is I jumped in and read every paper I could on everything ever published on Angelman syndrome. And what I learned was that there was incredible scientists doing incredible work on Angelman syndrome, but it was a really great decade to be a mouse with Angelman syndrome. And it wasn't yet the right decade to be my daughter Quincy living with Angelman syndrome, or the 500,000 other kids potentially around the world that are living with this single gene disorder. And so what I did was I jumped in head first, and I was lucky enough to join the foundation, which had really a single mission, which was to cure Angelman syndrome. And I was brought in with open arms, and I realized that they were doing incredible and amazing work, and they were advancing science, and they were advancing gene-targeted therapies for Angelman syndrome. But what was also clear, it was that the roadmap to get from mouse to human was unclear, and everyone was waiting for pharmaceutical companies to do it. And that bridge from mouse to human was clearly the valley of death that was before us. And there was no money in order to advance drugs. And really, parents didn't know how to do that. And they shouldn't know how to do that unless they work for a pharmaceutical company. And so what I did was I took the task from the foundation to help create a roadmap to get from mouse to human. How do we get from mouse to human candidate and human candidate to first in human? And so I became their chief science officer. And through that, we were able to advance 6 therapeutic platforms from mouse candidate to human candidate. And our first platform was an antisense oligonucleotide. And we ultimately wound up starting a company as a foundation because nobody else was advancing these programs for gene-targeted therapies, and advanced that through an IND and into a phase 1 clinical trial, of which it has now been acquired by Ultragenyx. So they're advancing that program. In addition, we've advanced an AAV gene therapy, a hematopoietic stem cell autologous gene therapy, and now a CRISPR gene editor. And we have 13 other programs that are being advanced, all through the support of the foundation. So I come here not only as a parent of a child with a severely debilitating disorder who is beautiful and deserves every opportunity in this world, because she will never live an independent life. She will never speak a single word. She will never be able to do the common day things that most children and adults should be able to do without a transformative therapy in her life. And we have had to advance that as parents in an organization, bringing in experts around the world to do that and get that to children. So I've not only been involved as a parent and a drug developer advancing these drugs, but as figuring out what doses are appropriate, not only on the non-clinical side, as we get through IND-enabling studies, toxicology and pharmacology, but then how does that get translated into the human population? How do we guide families? As the chief science officer of the organization, families look to our foundation to explain to them and educate them, and we believe strongly in knowledge is power, on how we educate them on dose optimization in the clinic in a drug that you can give again, like an ASO or an oral therapy, right? Dose optimization there and dose titration, that is very important, but possible. But what about a gene therapy where you could be in a low dose cohort or you could be in a high dose cohort? What about a gene editor where there's no going back from that? What risk are parents willing to take? What outcome are they expecting? What is important clinical meaningful change in this population where any change is hugely important and that could change their life and bring them more independence? And these are the thoughts and the cares and what keeps us up at night as parents as we make these decisions and help guide our community through these decisions. And so I'm here as a representative of our beautiful community who looks to our leadership to help them and help educate their neurologists, their PIs, and themselves in order to help make these very important decisions. And I'll also add that I serve as the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, which is a consortium of about 300 members, including industry, regulators, parents, foundations, and clinicians, working to develop endpoints and biomarkers in order to truly show what doses are effective. We can show what's effective in mice. It's a little harder in a neurodevelopmental disorder to show what's effective and meaningful that's measurable, not just by a parent's voice telling you what's measurable and what's meaningful, but actually what's measurable on a piece of paper. And so we've been working really, really hard in order to be able to do that, which helps us when we think about dose optimization as well.
Great. Thank you so much, Allison, for sharing your lived experience and all of sort of your knowledge base as well. So next, I'd like to welcome Ned Weinshanker, Dr. Ned Weinshanker, co-chair of the Patient Impact Council of the MPN Research Foundation, as well as many other hats. So Ned, please feel free to give your remarks now.
Thank you. Yes, I am representing the NPN Research Foundation as part of the Patient Impact Council, but I'm also involved in a number of other activities at Utah State University and also in a couple of biotech companies. That's been my career. I live in Providence, Utah. I am a patient diagnosed with primary myelofibrosis back in 2018. So I've been living with this for 6 years now. There are about 20,000 cases in the US of myelofibrosis, so it is a pretty rare disease, not as rare as some of the ones that have been talked about today, but still pretty rare. I looked at this kind of like an out-of-body experience because I've been involved in companies running clinical trials, and I looked around for what's available for therapy for primary myelofibrosis, PMF, and found that most of the drugs, or the only drugs available, just dealt with the symptoms and not the underlying disease. But I quickly found one trial that seemed to have some effect on the underlying disease, and I entered that trial in early March of 2019. So I've been on the trial for 5 years and 8 months now, and just recently transitioned from the full trial to an extended trial. The closest location for this trial was UCLA, and I live in northern Utah. So it's been quite a burden to travel down there. At the beginning of the trial, it was every 3 weeks. After about 9 months, it went to 6 weeks. And during COVID, I was able to convince them to let me come every 12 weeks, which is what it's been since that time. I was very happy that this was a phase 1, phase 2 open label study because I really wanted to get the drug. I didn't want to take any chances. And I was very pleased also that they had several arms of the study for patients who were transfusion dependent and patients who were transfusion independent, and also those taking the new drug or a combination of the new drug plus an already approved drug. I ended up on the dual drug part of it. The problems that I had, well, first they did a PK study right at the beginning, They had put me on 125 milligrams of the study drug. And after a week, I went back for the PK study and they rapidly reduced it to 75 milligrams. So I was really happy that they were quickly looking at doses and blood levels. So then my anemia started worsening to the point where I had to have transfusions, but they kept the levels the same. The other thing that started happening was a severe loss of taste to the point where 2 years into the study, I didn't want to eat and I was losing weight. Things tasted awful or they didn't have any taste at all. And I was able to negotiate with the study group and have them reduce the dose to the minimum, which was 50 milligrams of the study drug. And very quickly, a significant part of my taste came back. So one of the things that I'm concerned about is in the clinical study, having either the local clinic or the CRO or the company really take a look at everything that's going on for the patient and look at possibilities to ameliorate some of these side effects. I'm also wondering whether the loss of taste is a biomarker for effectiveness of the drug, because over this 5.5, almost, more than 5.5 years on the study, it's become very effective. I have gone off transfusions. My blood levels are almost up to normal. My platelets for the first time last week were in the normal range. So it's working. But it would have been nice to adjust that level of drug early on in the study. I think I'll leave it at that, and I'll have more to say during the discussions.
Great. Thanks so much, Ned, and really appreciate you sharing very much your lived experience and some of the opportunity for dose optimization and really ensuring, patient quality of life in other ways as well. So next, I'd like to introduce Dr. Justin West, co-founder, president, and director of clinical medicine for the KCNT1 Epilepsy Foundation. Justin, please feel free to go ahead and do your opening remarks when you're ready.
Yeah, Justin's just fine. So I'm Justin West. I'm a surgeon in California, and I'm involved in rare disease because of my 7-year-old son, Andrew. He has a KCNT1 mutation, which leads to a catastrophic DEE. So the seizures that our kids have are totally drug resistant. He's never been on fewer than 5 drugs concurrently at any given time. And they talk about developmental delay, but let's be honest, it's developmental arrest. My kid is 7 years old and still a functional 6-month-old. There's really no development. These kids also have anywhere from a 25 to 50% 10-year mortality. We're still working on figuring out the numbers, but it is, this is cancer, essentially. I mean, very high risk disease. And these are families that will take on a very high burden of risk. I would just say, with rare disease, kids like Andrew, they take over, our lives. We modify our house around them. Danielle, you talked about invisible ramps. I have actual ramps in my house. I mean, we moved out of our house for 6 months when Andrew got to the point where he was in a wheelchair because we cannot physically, as 50-year-old parents, carry him around our house anymore because we're putting him at risk and he's putting us at risk. So the evidence of Andrew's existence is just deeply imprinted around our entire house. We live our lives around our child. We've got a nanny in the house basically 5 days a week. We have a nurse who's essentially here 5 days a week. Our house is like a little mini hospital. Everyone in the family is, of course, in therapy, our kids, ourselves. I mean, it really dominates your life. My wife had to quit her job as an ophthalmologist to focus on keeping my kid alive. Andrew is only here because of the incredible dedication that my wife put into him. She was out of the workforce for 2 years and now is back working for the FDA in the device section. For me, I basically put my practice on hold, which has been financially catastrophic. But, at the end of the day, I decided to start a foundation, much like other people in this room. Allison, you're hard to follow because the way you share it so eloquently, but the same concept is how do we take what we've learned as problem-solving surgeons and apply it to a disease that nobody seems to have a roadmap for, and people are taking it on bits and pieces. How do you do it really coherently and aggressively with a fire lit because we have kids who are dying? 7 years ago, we had no therapies and maybe 1 or 2 drugs that were a very early phase discovery, and now we're on the verge of having 3 IND submissions in the next 6 to 12 months. So we are making progress, but we still lose a kid every month, which is totally unacceptable. We do have experience with dose finding, both with an ASO as well as for a repurposed drug that we found in our repurposing screen. I've seen the consequences of starting doses too high and advancing doses too quickly, but we've also seen the consequences of starting doses too low, where parents or patients don't get the positive feedback that there could be a signal of efficacy, and then you get people dropping out of trials that could potentially work for them. I think that one of the themes is, we've talked about paternalism here, and I think that part of paternalism is assumption making. As a surgeon, I do things every day. Every single thing I do hurts a patient. We have this concept of do no harm, which is so old school and ridiculous. It should be let go and put into a grave, because everything that Alice and I do to patients hurts them, right? There's a very high, complication rate, infection rate, mortality rate. And so I think the key is really it's proper education. I tell my patients my job is to cut and sew, but really it's about educating and empowering the patient, the individual patient, to make a decision for themselves if a therapy makes sense. And I think that we should probably not bucket people into they accept risk, they don't accept risk. When you don't, it should really be done on an individual basis, because the way, Allison accepts a risk should not influence in any way the way my family gets to be sort of labeled as the risk that we would accept. I think we have to do that. Each patient should have the opportunity to sort of share that and be educated by their physicians. I can tell you as a physician in clinical medicine that physicians do a really poor job educating. I think that, one of the other things is that we make these assumptions that we understand our patients because we have, sessions like this. Our drug developers think they understand our patients because they read all the papers about it. Our our clinicians assume they understand our patients because they're managing a disease. And then you watch how they set up, let's say, for example, a natural history study, and they tell that, families like ours with all the medical equipment that we are burdened with, that we're going to fly to the East Coast, to the Northeast corridor, where it takes 2 flights to get there, and you're going to travel with your pulse ox and your rescue meds and 2 adults, and this is the best plan that they can come up with to study us. Well, we're not even at the drug phase, but you're going to diminish interest because the hurdles that you're setting up are just absolutely uncrossable. So I invited pharma years ago to come stay at my house. I call it the 24-hour challenge. And I think that the onus should be on anybody in this landscape to really educate themselves better about the disease by virtue of exposing themselves to the families. So I now have CEOs who walk into my house, pick up my son, sit on my couch, push Andrew around in his wheelchair. And I think it's great. So pharma's doing it. I've had our clinician in the house. And I think, honestly, it'd be nice if regulatory folks, picked up that same invitation and showed up and learned about our families. Because if you're going to make decisions that either preclude us from getting drugs or allow us to get drugs, you should really understand, what our families are experiencing. I'll stop there because it might go on forever.
Great. Thank you so much, Justin, and certainly covered a lot of ground there in terms of your experience and all of the sort of challenging intersections in this space. So next we'll go to Julia Mauis, a patient advocate at The Right Dose. So Julia, please feel free to go ahead with opening remarks.
Thank you. So I live with metastatic breast cancer, and I lead the patient-centered dosing initiative, which is an initiative led by a group of us patients with metastatic breast cancer, and we're questioning the practice of routinely treating metastatic breast cancer patients with the maximum tolerated dose. So even in a disease like breast cancer, dosing is not simple and is not good in the way that we're currently in our current environment. So we did a survey of metastatic breast cancer patients. We surveyed over 1,000 patients, asking about their toxicity and their experience with their drugs, and we found that 86% of the patients experienced a significant toxicity. 20% of those patients have had to go to the ER from side effects of their drugs. Almost half of them, 43%, have had to skip a dose because of toxicity of that same drug. Basically, like these drugs are dosed, and the dose is determined based on these early phase trials that look at a very small number of patients for a very small amount of time. And then patients are expected to take these drugs for maybe their whole life, if they're lucky, right? Because when we run out of options is when we stop taking treatments. So we're really shining a light on dosing in both clinical trials and in clinical practice. Dose reductions are a very important tool in focusing on patients' quality of life, and that relationship and that interaction between the doctor and their patient is very important. Patients should be empowered to talk about their side effects, and using dosing as one of the tools to manage those side effects.
Great. Thank you so much, Julia, for sharing your experience as well. Really appreciate hearing some of the challenges you've highlighted. Last but not least, we do have Dr. Michael Pakanowski, Director of the Division of Translational and Precision Medicine in the FDA's Office of Clinical Pharmacology. So Mike, whenever you're ready, would love to hear some remarks and reactions.
Sure. Thank you, Victoria. I first have to thank all of our panel members here for sharing their stories. It's really important for me and FDA and all of us to hear this kind of feedback as we think about our approach to developing drugs for the patients and families that are managing rare diseases. And it's important to keep in view that extra layer of complexity that is introduced by research on these experimental medicines. I'll offer a couple of comments, but I'm more interested in hearing what the panel has to say, so I'll try and be quick. But our goal is really to make sure that we're getting safe and effective medicines to folks in need, and not getting the dose right can have a lot of negative consequences. One of which is failing to show that the drug actually works. So in terms of the day-to-day, we're working with companies to ensure that they put together a data package that really establishes how much drug is needed to produce a benefit without causing additional complications that can adversely impact quality of life. And this is particularly important when we have no treatments that are available, and we're really looking at the promise of drugs that can modify the underlying cause of a disease. So, first and foremost, it's critical to find any dosage that's effective. And we aspire to make sure that we can have an optimal dosage for each unique patient, because everyone's circumstances are different. Rare diseases programs are not conventional. I think the differences are familiar to most of the folks on this meeting. So perhaps it suffices to say that, all manners of evidence really need to be examined very carefully. And we're selecting single doses that are expected to work in what is usually a single trial. And that's usually done based on animal studies and the first human experience in either healthy subjects or patients. So there's really a critical need for animal models that are reliable and robust, as well as sensitive biomarkers that are hopefully clinically meaningful to be used in those initial ascending dose studies, because there is a critical time window to understand what dose is actually producing the intended effects. In addition, we also tend to recommend carrying forward more than one dosage into later trials so that we can see the effects of a range of different drug exposures, and incorporating these comprehensive assessments of drug exposure and biomarkers and other measures really allows us to evaluate differences in response based on how much drug gets into the body. And assuming the drug is effective, we can then consider different dosing approaches than might have been studied, including patient-friendly schedules, hopefully, as well as dosages for unstudied populations and tailored dosing for different subgroups. So our panel members, I think, have brought up a number of points that deserve some attention when it comes to getting the right dose. The value of this work, I think, comes through clearly in Ned's story, because it took a dose change to find the balance between an important side effect, but preserving the benefit of therapy. And, absent the ability to do trials where we're titrating the drug to an effect, which is really only suitable if we know what we're targeting, and that's often not the case in rare diseases. Having these data on multiple dosages really helps us establish where the floor and ceiling might be, so that we can understand the impact of those dose adjustments on the benefit risk profile of the drug. I also said before that we rely on comprehensive assessments of drug exposure and response, and we do face attention in rare disease drug development between the reality of living with the condition and the need to obtain data. And my personal perspective has always been that with so few patients available for studies, we should really be obtaining as much data as possible from each participant and treasure it. And this would mean, collecting extensive blood samples, tissue biopsies, scans, physiologic measures, and what have you. But we do have to appreciate that people can't tolerate this amount of poking and prodding, and there needs to be a balanced approach that makes sure we get the minimal information necessary to inform dosing, but also that we use all the information we do collect to the fullest extent. Another general consideration that was raised is, the patients often have to travel to participate in studies, and I'm excited by the prospect of bringing trials closer to home through local labs, local healthcare providers, telehealth, wearables, other sorts of technologies. We've issued guidance on this topic, decentralized trials, and hopefully these efforts should facilitate gathering that data in a less burdensome way and perhaps even encourage enrollment of more patients than we would ordinarily study, so that we can incorporate these designs that have multiple dosages. So I think we've been open to innovative solutions and really encourage drug developers to bring those forward for discussion. We understand these approaches often require more patients to be studied, more significant resources to be invested, but I'm confident that investment is worthwhile in the long run for patients. So,
Great. Thank you so much, Mike, for sharing those sort of thoughts and reflections on what we've heard so far. We've covered a lot of ground already. I know we don't have a ton of time for discussion questions today, but I do want to take a moment to turn to a couple of discussion questions we have prepared, as I think it'll be a nice continuation of conversation. So we do have a first question here for folks on how has clinical experience and or patient preference shaped administration practices for the different therapies utilized in treating rare disease populations. We've heard some of this, but we'd love to hear if folks would opine on what can we learn from these examples as it relates to opportunities for dose finding and optimization for treatments targeting such populations. So I did see Ned's hand up first. Ned, giving you the opportunity to share some brief comments if you wanted.
Yeah, just one of the things that the Patient Impact Council at NPN Research Foundation is doing is making the members of the council available to companies running clinical trials or planning to run clinical trials. And we have been able to review protocols before the trial begins and give input back to the companies from a patient perspective. I think that's a really important aspect, and I think that could be a general procedure that perhaps all protocols should be looked at from a patient perspective.
Great. Thank you so much, Ned, for sharing that. I saw Danielle jump in next. So Danielle, go ahead.
I just wanted to share something that we've experienced. We go to a wonderful children's hospital, and because of these challenges that we have with Charlie sitting for a blood draw, for example, we have to bring in a big person to come and, kind of help us with him and such. His team is very proactive in thinking about studies when he's having a standard of care visit. So when he was having his epilepsy surgery, they said, let's see if we have a study that might, require, blood draw while he's under anesthesia, for example, or something like that. And so thinking about how frequently people are already in the hospital getting a blood draw, for example, there's ways to piggyback on those procedures to reduce the burden. And so I just wanted to share that with you.
Great. Thank you so much. Any of the other panelists interested in jumping in on this question before we move on to the next one?
I had another comment for Michael with respect to distance participation. During the main part of my trial, I had to go to UCLA because they wanted to do MRIs on the same machine. They wanted to do bone marrow biopsies on the same. I had 26 MRIs and 8 or 9 bone marrow biopsies. Now on the extended study, the only thing they're going to do is a blood draw. And they're still requiring me to come to UCLA for that. I could easily go locally or even to the Huntsman Cancer Institute here in Utah, an hour and a half away. And right now they're saying, no, you have to come back to UCLA for this. I fly down, I go in, they take a blood sample, I see the nurse practitioner for 2.5 minutes and I leave, and all that travel for nothing, basically. So loosening the requirements and letting the companies do more distance evaluation, especially when it's pretty standard testing, is really important. And because there are so few patients, you can get more patients to participate if you made it easier. The companies made it easier and the rules were easier.
Great, thank you so much. Allison, please feel free to jump in.
I'll make this really brief because I know you want to get to the next question, but I would also add, coming at this, because I have the industry hat, the parent hat, and the foundation hat, and I'm gonna be wearing the foundation hat for this entire meeting. But I would also say, from a foundation perspective, in rare disease, particularly rare disease in which blood draws, EEGs, CSF sampling, any of these diagnostic tests are challenging, to say the least, with restraint. PTSD, where kids don't want to ever do an EEG again because they need to be restrained, wrapped for 45 minutes, and then they pull it off. So parents lay in bed with them all night, holding their arms down so they don't pull their EEG off because they need an overnight EEG for some of these trials. I personally believe it should be a requirement that all sponsors share baseline data in some repository in which any other sponsor can utilize it, because that would decrease the number of patients that would need to be in controlled studies. Bayesian statistics could be utilized and borrowed from with these baseline, well-conducted sampling, and including extra blood draws, extra CSF, EEGs. All of these are burdensome to families, but also require increased numbers in each of these trials when you have competing trials in order to be able to statistically power each study. And so if we can borrow from other studies in rare disease specifically, you can decrease patient burden quite tremendously if there was more sharing and everyone didn't hold everything as proprietary as they generally do. I understand the reason on the industry side to do that, but what I could tell you is the burden to families is tremendous. And if you can lower your numbers by borrowing other people's baseline data, and they can lower theirs by borrowing yours, it would actually change the field.
Great. Thank you so much, Allison, for those comments. This second question we have, I know a number of you have already touched on in your remarks as well, but just wanted to give an opportunity for folks to share additional thoughts here. So what are the considerations that you weigh when determining whether to participate in early phase trials of an investigational drug, where different dosages are being tested to determine which is optimal? What challenges or barriers may impact willingness or ability to partake in such trials? So I know that folks had shared some more general thoughts there, but would love to hear specifically towards the dose optimization and dose finding activities, if there are any additional considerations. Go ahead, Allison.
Yeah, I can certainly take this one to start, just because we have 4 programs in clinic and 3 in IND enabling approaching the clinic, most of which are gene targeted therapies. But only 1 is dose escalation, and the rest are going to be gene therapies, which essentially are going to be irreversible. And so really, what the questions families ask, as I wound up fielding many of these and really having conversations with neurologists, clinicians, families, you know, in public forums, and we even do surveys of patient preferences and patient understanding, and trying to really get a sense in your head around what families do understand, particularly when it's a pediatric heavy population, and these are nonverbal children or adults living with these disorders. They cannot make these decisions for themselves. So these decisions are being made by parents and caregivers. And so what is very, very important to our community, which I'm so proud of our community for, is their ability to have access and exposure to data. Our parents are sophisticated, they're incredibly smart, and they should never be underestimated in their ability to understand data. And what they want to know is, what did this look like in a mouse? How did you make your dose selection based on those proof of concept studies in mouse? When they have to compare, do I want an AAV gene therapy or a CRISPR gene editor or an autologous ex vivo hematopoietic stem cell gene therapy or an ASO, in which we can dose advance and we can escalate over time and find that optimal dose? The questions they ask are really important questions, which is, what did this look like in the mouse and how do those compare? Not that a mouse is a human, we know they're not, but that's all we have sometimes when you're going at this at a phase 1-2 level, is how did those compare? How am I as a parent and our family willing to go back and forth for redosing every month, to every 2 to 3 months, maybe every 4 months over the long term. Patient access to hospitals to be able to be in a study in which you're redosed versus being in a study in which it's a single dose and then it's just monitoring that can be done remotely really impacts choice for families. And then in addition, really understanding the history of adverse events with different therapies. There are a lot of publicly known adverse events, but many times with some of these therapeutic modalities, adverse events are unknown until it's that transgene or it's that sequence or it's that drug cocktail in which you might appreciate or see an adverse event. So really educating families about what adverse events are known for the class of the drug or biologic and what are potential adverse events that they need to really consent to and really be explained what those can be and what risk they're willing to take. And I would say that the biggest risk families are willing to take is very independent to that family. But what they really want to know is, I'm willing to take a risk most times. It's just a matter of, can I get to a hospital every 3 months or can I not? And if I can't, am I willing to be in a low dose or a high dose cohort? Am I willing to be in low dose and take that risk of being underdosed? Or am I willing to be in a high dose and take that risk that it's toxic? And when it's irreversible, those are the risks you're taking, is lack of effect because it's too low or adversity because it's too high. And those are, in some people's minds, equivalently risky, right? So you really need to have that conversation. And to Justin's point, not just with your local neurologist, with your clinician. They don't always or even usually understand drug development. You need to have that conversation with the PI on that trial who has been through the IB and been through the ICF and understands the data behind it to really explain to parents to help them to make the best decision. We as a foundation educate them on the highest level of understanding animal data and the potential that that could translate to human data and the potential that it can't. And it really is knowledge is power, in my opinion, with all of this.
Great. Thanks so much, Allison. And I know Ned and Danielle, I see your hands raised, but just want to give an opportunity for Justin and Julie to jump into the conversation as well, if you have any thoughts on this one or the prior question as well. Go ahead, Justin.
It's hard to follow Allison. I mean, she as usual, she nails all the high level points and every point in between. So no, I mean, I think really it's for the simple thing I would just say is that's come up recently as we're working on clinical trial readiness in our community is the burden. I mean, we have families that are so chronically exhausted, like we will step over trash in our house 15 times throughout the day, even though it takes 2 seconds to bend over, pick it up. So every single hurdle that we add that could potentially make it harder to participate in the trial is gonna be one less family that participates. So I think that remote trials, decentralized trials, I think should always be a focal point when the drug class makes sense. Checking in remotely, Zoom visits, just recognizing that we have all these technologies that and at the end of the day, kids like ours, they're the least important person in the room. I mean, all of our decision making is between the physician and the parent, and that means it's a conversation, which means it can almost always be done remotely. So I think just reducing the barriers in terms of doses. Yes, I mean, Allison hit the super critical points. Totally agree. I think we just have to focus on these parents are exhausted, and asking them to do one more thing, even if it's a drug that could change a life, rescue a phenotype. It's incredibly. It's shocking to me how hard it is, but I also totally understand it being an exhausted parent.
Great. Thank you so much. I know we're going just a couple minutes over, taking moderator's prerogative. So I'll turn to Ned and Danielle next, and then do one more round through the remainder of folks. So just before you jump in, Ned, want to just leave folks with one last question for you to respond to, if you'd like. If there is a sentence or two you would like to share to really highlight for the panelists for the remainder of today's sessions, for them to opine on, think about, or respond to, please feel free to add that into sort of last comments here. So Ned, I'll turn to you.
What I was going to say is appropriate for that last comment. There are, for MPNs, there are 4 JAK inhibitors, Janus kinase inhibitors, that are approved. not one of them has an efficacy for more than 25 to 30% of the patients, maybe 35% of the patients. And yet we have this amazing amount of data from the clinical trials about the genetic profile of all of these patients. There should be a way to bring all of that information together and see if there's a pattern, why one drug works with a particular pattern of mutation and another drug doesn't, so that we can be much better in prescribing a drug for a new patient based on the genetic pattern, if that turns out to be the case. So I think that's a major issue, because this is a very heterogeneous disease. No two patients describe it the same way, and it's probably due to genetic and other situations within the patient. So
Great.
pooling that information is really critical.
Wonderful. Thank you so much, Ned. Danielle, I'll turn to you next.
Yeah, I just wanted to say that, you know, what the life Justin describes, that's my life too. And a lot of our lives. I didn't emphasize it quite like he did, but it really is what you, if I have to schedule an MRI on the phone, I can't do it via email. I'm so anxious about it. I just going back and forth on just one more thing. I cannot do it. It's so hard. And there's a stamina issue after 14 years. I'm just exhausted, and I have other kids, and I have a job and everything. So never underestimate the role that plays, how just exhausting it is. And from a practical standpoint, the question just now, what would the last question, what would you want to know, or what would go into your decision to participate? I'm not wearing any hat but just the mom hat right now. And practically what I would look at would be the questions on the data that you'd ask me to collect at home. Because oftentimes I feel like the questions are not, like, you really don't seem to understand what is going on. Like the seizure diaries, for example, don't represent the types of seizure events or strange events that we see. And so something that would influence me would be, do I think that the data that I'm collecting are actually going to be useful? And that might make me think, I don't know if this is going to be worth our time, because I think you're collecting garbage, frankly. And I've had lots of seizure diaries that people have made up that didn't make any sense to me, and I didn't feel like would really capture it. And they'd say, do your best. Right? It would be something for Charlie to answer, but he can't answer. So they'd say, well, just do your best. No, you can't. That's not good enough, right? Like that's not worthy of all the burden and things that Justin had described. And so to answer the question, what would be the sentence? I would just say to stop and slow down and understand the condition, truly understand. You know, these diseases are devastating enough to put all this effort into studying and to developing this trial, then understand them at that same level. Understand that, you know, this is heroic. It's a devastating condition. This drug will, you know, totally change people's lives. Why is it going to change their lives that much? Because they're devastating. And so it's more than just that one symptom or that one seizure. It's everything all around it that goes into it. And that's both what makes it devastating, but also what is so exciting about research in this field. Thank you.
Great. Thank you so much, Danielle. Julia, if you'd like to go with any quick hits.
Yeah, we think things are very hard in a disease like metastatic breast cancer, and the way we are given drugs and we are required to participate in clinical trials, and we see that trial participation rates aren't great. And then we hear stories like the families on this call and realize that obviously it can be so much worse in terms of access, and we're so far away from really designing trials and designing research that is easy for people to participate, not to mention the huge layer of equity that comes on top of all of this, because the people that I'm hearing on this call are bending over backwards to make these research participations happen. And they're privileged to have the resources and the access that they do. And that is not the reality of the majority of patients out there. So in the end, the data that we're going to have is going to be very skewed. It's not representing the whole population of patients experiencing each of these conditions.
Great, thank you so much, Julia. Okay, Justin, Allison, Mike, one last sentence you might each have to share with us.
I'll go. I'll go, I guess, if Justin, you're not going to go. Justin and I will just feed off each other. So you can go before me or after me. So I would say one sentence is to understand these decisions need to be made by the families and patients themselves. And what a regulator or a sponsor might believe is the right answer is often not when you're faced with living with these disorders. And knowledge is power. So parents and patients need to understand the data to make educated, strong, and rational decisions. And if they can't understand it, they need to be guided by the PI in a way that is clear and educational. Because the risk families are willing to take for devastating disorders should never be underestimated. Even if it is not a deadly disorder, if it's this debilitating, it is absolutely devastating to not only the patient's life, but to their families. And that shouldn't be underestimated.
Great, thank you so much, Allison. Go ahead, Justin.
Yeah, I think that just with ultra rare disease, since that's our neighborhood, just needs to be looked at differently. I think that with regards to the standards or the data standards that you're going to set or the outcome measures that you're going to set, I think it's one thing if you're developing the 25th drug for hypertension, then and ultimately going to bring to the market some drug that has a fractional improvement in hypertension control. That drug should necessarily meet an incredibly high level of safety and efficacy standards if it's going to be the next one in a pile of many. But when you have serious unmet needs where you've essentially lost a life. I mean, Andrew doesn't have a life. He has nothing, doesn't have a life approaching a human here, and a high risk for mortality. I think the bar needs to necessarily be lower. And quite frankly, I think it shouldn't be too dissimilar to the clinical bar. When we change drugs for Andrew in the clinic, we don't ask for how his biomarkers respond. We ask for how does his seizure burden improve, because at the end of the day, that's what we look at. And I don't think that the outcome should be really any different. We've had companies go to the FDA and get rejected because they don't have a biomarker. Well, no, most diseases don't. Certainly most diseases in epilepsy and rare disease. So I think that we shouldn't really be looking to hold us to a higher standard when the hardest job should be developing a drug that works. It shouldn't be we shouldn't be going beyond that, setting limits higher for the burden of proof, and the burden to participate should not be the rate limiting step. It should really be the hard science. That should be the hardest part. And sometimes it seems like it's everything else that's harder. And that just seems totally unnecessary.
Great, thank you so much, Justin. Mike, do you have one sentence you want to leave us with?
No, I'll yield. Thank you.
Okay, great. Thank you so much. All right, I really appreciate everyone's time on this panel, and really appreciate folks letting me use moderator's prerogative. As we can see, the patient and caregiver voice is so critical to this conversation. And just wanted to know we got some lovely comments in the chat and Q&A from some other panelists and attendees about how they deeply appreciate folks sharing their lived experience and reflections on what's needed to, sort of really move products forward in the space of rare disease. So with that, thank you again, everyone, and I'll turn it over to Nancy Allen Lapointe for the next session.
Great. Thank you, Tori, so much. And thanks to everyone in that session. I mean, that was actually a wonderful way to start things off. So thank you. As mentioned, my name is Nancy Allen Lapointe, and I'm an adjunct associate professor in medicine at Duke University and faculty fellow at the Duke-Margolis Institute for Health Policy. And today I have the privilege to serve as moderator for our second session, entitled Early Phase Clinical Trial Design to Support Dose Finding and Optimization. In this session, we'll be exploring novel statistical methodologies and trial approaches to support dose finding and optimization in early phase trials, and discussing the role of non-clinical models in informing early phase trial design. And certainly, as I mentioned, session 1 has provided an excellent framing of the critical and important urgency of more rapid and effective development in the therapeutics for rare diseases and the importance of dose finding and optimization. So with that, we have a fantastic, I apologize, I think my camera is having some trouble here. I apologize for that. We'll try to get that reset. Apologize for the delay. Okay, I'm going to proceed without the camera momentarily so we can keep things moving. We have a fantastic group of experts with us today to provide some presentations as well as opening remarks, which will then be followed by a moderated discussion. So to start things off, we'll first be hearing a presentation from Dr. Mark Rogie. Dr. Rogie is an adjunct professor at the Center for Pharmacometrics and Systems Pharmacology at the University of Florida. So Mark, go ahead and take it away.
Well, good morning, everyone. What a great session we just heard here. I wish it actually could have gone on a bit longer. Hearing, first from Isam on where FDA is looking at rare disease and how we ought to be moving forward. Mike Pakinowski's comments, just I think really spot on with where we are trying to go on the industry side. And as a little background, 30 years or more now, I've been working in drug development in pharmaceutical industry, as well as now being on faculty at the University of Florida. And rare disease has really been one of my passions. I believe it was Danielle made a comment, right at the end of the last session there around understanding the disease. And once we understand that disease, and by the way, we're now start going into the details. The devil is in the details here in this session. Understanding the genetics, the epigenetics, the intrinsic and extrinsic factors that affect the onset and the rate of progression of the disease. I don't think that has been given the attention that it deserves, and certainly now industry, I believe, is beginning to recognize it, but hearing from FDA and others, and I think that message getting out, much more emphasis is going to be put on the actual human element of this disease, as opposed to, say, for example, animal models. And you're going to be hearing me over the next 10 minutes or so downplay a little bit the animal models, because again, consistent what was said in this last session, I think we all want to see these products, if there's confidence in them, to move forward into clinical trials as soon as possible, as fast as possible. And getting to Justin's comment as well that he made also at the end there, we are focused a lot on biomarkers. If those biomarkers are feasible, they should certainly be developed, but they shouldn't necessarily stand in the way of getting that product into humans. I'm going to give an example at the very end here, and I think it actually illustrates that very point. If we could go to the next slide, please. So probably what I can start off by saying here initially is, what is it that we should not be doing? Let's not set that bar inappropriately high. I think we were hearing that over and over again in the last session. That R&D, research and early development, needs to provide confidence that, that human investigation is warranted. It can proceed with controllable risk. Are we able to modulate the pathway, potentially have some significant benefit in the disease? We'll learn that when we get into human trials. What's important in that research phase, that early development phase, before we get into humans, is the best candidate being chosen. Does it have the activity that it needs to have? Does it have the specificity? Is it not promiscuous and affecting other pathways where we now may begin to have some rather serious safety consequences? And the delivery, is it actually getting to the site of action and staying there for a significant period of time? And then, of course, throughout all of this, and this is really the major focus that I see in that preclinical stage, is acceptable risk. And I'll touch on that in another slide here. But can we go into human trials and have a controllable, acceptable risk? There's always going to be some risk, of course, but not to stand in the way, particularly in a rare disease stage where the consequences of accumulating more disability, usually in an irreversible manner, far outweighs any potential smaller safety risk. So next slide. So, for this, I'd say, the team requires some focus on some specific elements, milestones that ought to be, really first and foremost in how they want to plan to move that product forward. We do need to understand, are there biomarkers that could be used? Of course, diagnostic, and I think we all understand that those can be very important. We need to know who has the disease. We certainly want to get the right patients in the trials when a product is ultimately going to be developed. But disease progression markers, again, if they're available, and certainly some kind of pharmacologic marker if it's possible. And that's not necessarily a chemical biomarker, something in the plasma or CSF. It could be an imaging biomarker too, for that matter, at least in those very early trials. What tools are most important here? We have this remarkably large toolbox of ways of looking at products, and I think sometimes because the tools are there, we feel we need to use them. It's a wrong way of looking at it. There is an asymptotic improvement in value, you might say, in understanding your product as you begin to do more and more of this work. I think we need to do a better job choosing what we're going to look at initially, and then ultimately move as fast as we can into those early clinical trials. You can see at the very bottom on the left side of that slide, limited animal model studies, and this is really on the research side largely, only when justified. I guess in my own experience, probably for every animal model that I've used, there's been 5 that did not work, and that's not a good return on investment. I think we have other tools now, particularly iPSCs, microphysiological systems, other means of knowing whether or not we've got a good chance of the drug doing what it intended to do, and does it get to the target. And then if there is safety, which is more on the right side of this slide, if we can demonstrate that there's some type of controllable or acceptable safety risk, that we get into human trials as soon as possible. And again, I think this gets to some comments from the earlier session. We go in at pharmacologically active doses, low pharmacologically active doses, if that can be determined, being assured at the same time that there is an element of safety there that's not going to outweigh any potential benefit. Next slide, please. So Justin brought this up I think in his last comments there. We've almost got a bit of a paradox here, in this preclinical thoroughness of what we're trying to do. There's conflict with both speed and caution. We can harm through optimism if we don't understand the consequence of a new modality, a new chemotype of an antisense oligonucleotide or whatever it might be, going into humans too fast, and now there is some consequence that only adds significantly to the burden of the disease that patient already has. However, and I would say I think that's likely going to be a very rare event. There's harm through caution. I think that's maybe what Justin was getting at. We cannot hold ourselves back thinking that some small additional bit of information of questionable value is going to help us make a decision or result in a safer clinical program later. We have to think about it. We validate all of this preclinical work by doing human trials. We often don't know unless there's precedence, of course, we don't know the value of these products until we actually, or any results we get from any of the work we're doing with these products until we actually get into human trials. So it's on our shoulder, it's our burden that we need to move as fast as possible, as smart as possible, but doing it to ensure that at least no significant or uncontrollable adverse event should get in the way. Go to the next slide, please. So how much knowledge then really is necessary, to get into trials? I think is important, and as we move from sort of the left side of this table to the right side, demonstrate that the molecule, the therapeutic, whatever it has, happens to be, has sufficient affinity for the target or activity, that it gets to the site of action, remains at that site of action for a sufficient period of time. But you can also then develop sort of the translational element to this, and Isam was getting to this in the very end of his presentation. We need to aggregate and pull together all of the data from the individual studies that are conducted to create a story, an interpretable or understandable story around what happens if we increase dose, what happens if we decrease dose. Ned made a very good comment in his treatment where he had this terrible taste that was really beginning to affect his ability to continue with the product, potentially through some physiologically based modeling, or even better, some quantitative, systems pharmacology modeling. We could understand what is going to be the consequence of lowering that dose. How much improvement could there be in the adverse event profile, but what would the hit be? What would the consequence then be on the therapeutic activity of the product as well? These are not sequential activities. These largely can fit in where it's best for them to fit in, and they, again, should not be standing in the way of starting clinical trials. But certainly as enough data become available, you would want to begin to do more and more of these integrated models. And I think Raj will be talking a little bit later. We'll be hearing from him and all the really exciting work he's got going on at FDA in more model-informed drug development. Could go to the next slide, please. So let me just finish off with a quick example here. I was really fortunate a few years ago to work at Biogen with a team of colleagues also from Ionis in a rare disease called spinal muscular atrophy. And for those that aren't familiar with it, it robs infants in its worst form of their ability to use their limbs, and then it goes on to unfortunately rob them of their ability to breathe. Those with the worst form of the disease, SMA type 1, generally have a lifespan of less than 2 years. The antisense oligonucleotide it actually came from the creativity of Frank Bennett at Ionis and some other people. Were able to take a very similar protein. In SMA, the SMN1 protein is deficient. It doesn't exist, or at least it's non-functional. There's a similar protein, a homolog that we all make, SMN2, that unfortunately is spliced naturally. Frank and his colleagues came up with an antisense oligonucleotide that could prevent that splicing. The SMN2 then retained relatively sufficient amount of activity to then prevent this disease, from occurring. We were fortunate here with this product in that there's a mouse model that highly recapitulates what happens in humans through a fair amount of in vitro work and then work with this SMA delta 7 mouse model. We were able to understand what types of exposures in the mouse would then ultimately lead to uptake into spinal cord and brain where the ASO needed to go, and then prevent this splicing event from occurring, preserving the SMN2 activity, and then ultimately preserving motor neuron function and survival. We were also fortunate in having a wealth of information from the past in other diseases, in neurodegeneration, that when neurons are lost, when they die, something called neurofilament light, it's a component of neurons, is released. It ends up in CSF, but also in plasma. and we were able to measure neurofilament light, and that work is continuing on. In fact, I can show you in the next slide, please. Thank you. And I'm not going to go into this because I know I'm running out of time here, but some of the biomarker work that we were able to use both in our animal models, but then ultimately in humans is shown on this slide. And the ASO from the Western blot in the top left, you can see with nusinersen, the ASO, we're able to completely recover the SMN protein in these animals. The bar graph to the right demonstrates that motor neuron function was preserved, and then even function, in the bottom two figures on the left side, you can see that animals that SMN delta 7 animals that otherwise would not survive continued to gain weight and thrive just as the control animals did, and they were able to regain their writing reflex. Now, over on the right-hand side, this illustrates neurofilament light, and while we did not have this data here at the time to help us make some of our early clinical decisions, it illustrates what neurofilament light looks like, particularly with the red line. So the type 1 patients, very, very high, in excess of 10,000 picograms per mil in CSF. However, with nusinersen therapy, literally within a few months, it's brought down to near normal levels, which is that horizontal gray box. So early on at the time, while we did not have the ability to look at it this thoroughly in our nusinersen program, soon after we were able to, and it continues to be done so now. And by the way, too, as you can imagine, the function and survival in these kids was remarkable. They went from being just in having a terrible prognosis to literally now being able to move on to a relatively normal life. Next slide. So just finish up here, very quick. And I think this gets to a bit of what Isam was talking about at the very beginning. In I think it was on his last slide, actually, where we need to integrate our data and we need to make it translatable. And one of the smartest and I think most appropriate ways of doing that is creating more mechanistic, even semi-mechanistic, if that's what can be done, models that relate to where the drug is going in a more defined or limited fashion across different tissues. But also what it is doing then when it gets into those tissues, and then under different dosing paradigms, other conditions, we can understand what type of dose is going to be necessary to achieve, the outcome we want. This is the semi-mechanistic model that we created for nusinersen, for ASOs. It's been used outside of SMA. It continues to be done so for different brain diseases, but it's had just a tremendous amount of value. And I think what's also important here in all of this did not require hundreds of animals and other input that could have slowed us down for potentially years to get to the point of having a useful model. It was from a very limited number of non-human primates that we used in our toxicology program, and certainly some of the data that I showed in the previous slide from the SMN delta 7 animal. Next slide. So immediate value, and I just want to focus on this model-informed development at the end. I think we should all be moving towards this if the opportunity permits. Tremendous confidence in our dose responses, our dose frequencies. ASOs, by the way, at least for CNS, are often given, CNS disease is often given intrathecally, very invasive, some risk. So giving them as infrequently as possible is ideal. This also gave us the opportunity to look at younger patients. We started out in kids that were a little bit older with the disease and had less severe disease, but we really wanted to move into the more severe patients as soon as possible. And these were the infants. So you can imagine their length of their spinal cord, the CSF volume, flow rates of CSF, All of these elements that go into this type of model were able to be looked at appropriately to help us understand the most appropriate dosing then in those kids. So I think I'm going to leave it at that because we're going way over time here. We'll have some time during the discussion period to maybe talk about some more elements of either this example or some of the other things that I've been able to work on. But again, I thank everyone's time and I look forward to the discussion.
Great. Thanks, Mark. I know everyone is very passionate about this topic, so it's wonderful to hear these presentations. Next, we're going to have a presentation from Dr. Nicholas Shork. He's a distinguished professor and co-director of the Division of Clinical Genomics and Therapeutics at the Translational Genomics Research Institute. So, Nicholas, take it away.
Okay. Hey, this is a very inspiring meeting and happy to have been asked to make a few comments. I'm going to embellish and maybe extend some of Mark's comments, and I'll try to be brief. But there's a few things that I'd like to touch on. Here are the themes. I have 1-2 slides for each. What are the goals of early stage trials for rare disease? I mean, some of this has been discussed. I'll give you my own take on it. Talk about, as Mark did, some of the limitations of animal models and preclinical research as it's used to guide kind of studies for rare diseases. Then talk about some more academic things, but I think have real bearing on how we're going to pursue studies in the future, having to do with assumptions about mechanisms of action of drugs used to treat rare disease and trying to deal with the complexities of human biology. And then talk a little bit, I won't go through the theory, but some emerging designs that are referred to as single case experimental designs or SCEDs, N of 1 trials, as Isam had mentioned them. Next slide, please. So multiple goals, as we know, in very patient-specific therapeutic early stage trials. They're not just about dosing, they're also about safety, they're also about efficacy. So combining all 3 developmental phases into one study is really the problem and the goal for studies of rare diseases. But a couple questions in the left yellow panel that one could ask. Is the intervention part of a platform like an ASO? Could a trial using that platform approach inform other trials in biologically meaningful ways? This was brought up by, I think, Allison and Justin. Why couldn't we leverage information from the experience of other patients if, in fact, the therapeutic that's being evaluated is one of a type that is being tested in other people? Is it possible to exploit different biomarkers? I agree with what Justin said. The goal should be getting the patients healthier and not necessarily just focusing on whether a biomarker is modulated by a therapy, but biomarkers have a place in rare disease trials. Is there a reason to believe that participants in a trial must have an absolute minimal burden? Obviously, this bears on patient burden, as the panel 1 or session 1 speakers alluded to. And can the breadth of the effect of the intervention be anticipated? Now, this is important for the design of trials. So again, I'm an academic, but definitely motivated by a desire to help. But there are some design elements that we can discuss that I'll expose you to. I'll just point out a very recent paper that's available in a preprint archives by the Ionis and Lorem group on their experience with 173 patients that got ASOs. If you hit the spacebar, next slide, you'll just see that they published some tables describing how they went about preclinical research. This could be very informative for future discussions. There was another paper that kind of contrasted the preclinical evidence for a couple of different ASOs. Next slide, please. So what are some of the limits of preclinical models? Well, they're pretty obvious and in fact being recognized by the community. And I would argue, in fact, that non-human models are improving because we're recognizing their limits. So we know what situations they might be most appropriate for, say, a mouse model of some particular disease. They are very important for initial toxin safety studies, just to kind of level set. Is this therapeutic likely to do more harm than good in a living organism? They also can be used to establish dose response because it shows again that they can be done. Whether or not you could literally translate what happened in a mouse to a human in terms of doses, that's an open question. I have my doubts, But if you can establish a dose-response relationship in a living organism, it should at least provide motivation that you could do the same in humans. One thing that everyone recognizes, there are huge within and between species genomic differences and other types of differences that complicate the translation of preclinical models to humans. And cell models, which I also am an advocate for, have their own limits. Hit the next slide, please. So this wasn't coordinated with Justin's comments about antihypertensives, but I just use it as an example because there are many routes, therapeutic routes that one could take to try to treat the disease. That's in the green box at the bottom. For example, if someone has an elevated heart rate, you might want to give them a beta blocker. The problem is there might be other perturbations in the system beyond the one that you're targeting therapeutically that could compromise the benefit of the drug. This speaks to the need for seeing these early stage trials for what they are. They are empirical exercises that are very hard to anticipate with respect to the likely benefits of the drug. And this needs to be taken into consideration in the design of studies. Next slide, please. Some of the questions that I think about when designing studies are listed in this little 2x2 table. Is the mechanism of action of the drug in the leftmost column here very patient specific, like an ASO or a CRISPR therapeutic or something like that? Or does it tap into some ubiquitous and general mechanism? Another question is, how broad is the phenotypic effect of the drug? That's very rarely considered in early stage trials. Is it likely to have a very focused phenotypic effect or a broad phenotypic effect, like ASOs? As Justin and Allison and others alluded to, most rare diseases are syndromic. The mutations in a primary gene tend to have a very pleiotropic or broad effect that needs to be taken into consideration when designing studies with respect to the endpoints of relevance to the study. Next slide, please. So I'm just going to mention this. Again, this might be of more academic appeal, but there have been studies looking at how pleiotropic perturbations in genes are, and it turns out that they, for the most part, are very pleiotropic. That suggests that if you modulate a particular gene, turn it off, turn it on, do something about it, it might have broader ripple effects throughout the body that you'd want to know about, and those could be crucial for putting in perspective the likely benefits of a drug like an ASO to treat a more syndromic-like condition, like Batten disease or something of that sort. So the next slide, please. So my strategy is not unlike the one that Issam showed in some of his slides, which is to design more sophisticated studies that limit patient burden to the degree that makes sense. And this is a complicated discussion because you don't want to poke and prod patients that are already compromised. On the other hand, we need to collect the appropriate type of data, so a balance needs to be struck. I believe that balance can be struck with appropriate designs and greater patient and family engagement. So this slide is just more concept oriented, showing how one might be able to design studies, say do a natural history study followed by an intervention study where the response to the intervention is guided by what was learned in the natural history study. And all the statistical technologies, to the degree they make sense to incorporate, like blinding and randomization and stuff, could, in theory, be leveraged in such studies. Okay, next slide, please. So I'm not going to go through things in great detail, but there are other early phase studies that are used in other contexts that leverage strategies that I think can be used in rare disease studies. So for example, in therapeutic drug monitoring studies, biomarkers, a marker of activity, the level in the plasma of the drug, to the degree those can be measured in a convenient way in a patient, they should be leveraged because they might be better vehicles for establishing, say, causal dose response effects and whole body effects. So I do believe that some of the strategies leveraged in other types of early stage trials, like therapeutic drug monitoring, could be exploited in rare disease studies. Okay, next slide, please. So again, I'm not going to go through in great detail, just maybe emphasize that statisticians like myself, other people designing trials, are coming up with strategies that might minimize the amount of data that needs to be collected in any one trial, but maximize the insight in terms of the patient benefit. Next slide, please. And this has been discussed for some time. I'll just point out, not so coincidentally, there was a panel put together some time ago by Isam and colleagues almost 15 years ago talking about novel trial designs. I was fortunate enough to have been asked to participate. Next slide, please. Again, I'll just mention that one can design studies. I won't go through the math that might minimize the patient burden, but collect enough data to make compelling and valid statistical claims about the efficacy or tolerability or dosing of a particular drug. Okay, last slide. This is crucial and echoes the points made in the first session and some that Mark and Isam brought up. A lot of discussion on trials being fit for purpose. So it's one thing to pursue a trial for scientific benefit. It's another one to pursue things to benefit a patient. And I think those two need to come together. So I do believe clinical trials can be pursued in a way that engages the patient, and we can accomplish two things: get the patient better and also learn enough about the mechanism of action of a drug to say compelling things about its use going forward. So I'll stop there, and thanks very much.
Great. Thank you so much, Nicholas. Now we're going to bring up our remaining three experts for the session one by one for them to share their opening remarks. And these will just be a couple of minutes each. So first, I'd like to turn to Dr. Vikram Sinha. He's a global head of therapeutic areas at Novartis Institute for Biomedical Research. Vikram, take it away.
Well, thank you very much, Nancy. Just let me first at the outset just thank the organizers for the invitation and to participate in this panel. It is, of course, a topic that is of huge significance. I must say I came into this session with a slightly different view of providing comments, but the first session with the caregivers, the parents, the advocates actually put a different focus in the way I probably would think about those finding in early clinical development. Nevertheless, maybe I'll focus on a couple of things. Some of these points have been very well made, but just to re-emphasize, I think there is a real barrier with animal models. Dr. Rogi's actually stated this very well. The translatability is not always clear. And unless we have something really precedented in the field, it's very, very difficult to translate and be certain. The whole area, the big challenge is sample size, small numbers of patients. Often we don't have huge databases with natural history. And so the value of data from each patient becomes very, very significant. But on the other side, we do have very significant enablers. By this, I mean, we are starting to see great strides in and focus in trying to collect databases of natural history. Registries are going into place. There's tremendous work being done on digital endpoints, digital biomarkers, and other means of collecting information. Blood sampling, which now is really very, very important in understanding those response relationships, has moved from labs to bedside. And we have newer technologies that are being used in many such trials to reduce patient burdens. And then finally, we have many tools at our disposal. Dr. Rogi spoke about a few of them. We have the whole arena of pharmacometric approaches that allow us to understand and often interpolate and sometimes extrapolate for different situations. And also, I think, finally, the area that probably deserves the most attention to maximize this challenge with small n's is the use of adaptive designs. And I think adaptive designs using Bayesian borrowing is really the way to really think about getting a handle on not only efficacy, which I think is paramount, but also giving us a sense of how we understand, increase our understanding of the overall dose response curve. So I think these are the real approaches that are front and center. The good news, I think, is everyone is giving this significant thought. And the FDA is taking a lead in this. I think they're expecting this. I think when they expect this, they move sponsors in a different direction and make them really think about each indication, really think about each study design in a very different way. I think that's really the need of the hour. With that, I'll stop here. Thank you.
Okay, thank you so much, Vikram. Next, I'd like to turn to Dr. Shauna Weiss, who is the Acting Pharmacology Toxicology Supervisor supporting the Division of Rare Disease and Medical Genetics in the Center for Drug Evaluation at the FDA. Shauna?
Thank you, and thanks to the organizers for the invitation. So I've been asked to talk a little bit about how we use animal data to set doses for initial clinical trials. And so I'll briefly talk about dose setting for first-in-human trials, but then I'll go on to discuss very quickly how we use these data later in development. I'm sure everyone in this room knows that for first-in-human studies, the emphasis is more on safety. So we're interested in understanding the no adverse effect level and what the toxicities are in animal studies. And then we want to understand, ideally, something about the concentrations needed for efficacy in some system. And oftentimes that system is an animal model, like a knockout mouse or a knock-in mouse, but sometimes it can even be in vitro data or microphysiologic data, just showing target engagement and some sort of functional outcome. And so that ratio of, where we see toxicity divided by where we see efficacy, if you will, is the therapeutic window that we can explore pharmacokinetically during early clinical trials. And so it's really helpful to have identified a biomarker from some system, primarily an animal or some sort of trackable plasma biomarker or a functional outcome that would allow us to optimize the dose setting in early clinical trials, because as it's been pointed out, we don't want to dose too low because being subtherapeutic isn't beneficial, but dosing at too high of a concentration or a dose can lead to toxicities, both those that we predict in animals and those that we can't predict. So necessarily the toxicity data really define the dose range early in development. But fortunately, once we have early clinical safety data and we have some understanding of how representative those toxicities that we identified are in patients, we do have the ability to modulate the dose range if we're not seeing those findings in patients, and perhaps even exceed exposures that were found to be safe in animals in some cases, of course, depending on the safety signal that was identified. So I want to echo the findings of the previous speaker, particularly Mark and Nicholas, about the relative importance of animal data versus humans. Clearly, humans are more important. So having said that, though, animal data can be extremely important, particularly animal efficacy models, and that's true for a number of reasons. Determining the target dose or exposure level associated with efficacy allows you to understand both the therapeutic window and the pathogenesis of the disease, understanding how those pathogenic biomarkers or functional outcomes can be changed by the exposure over that efficacious dose range is important. Understanding the relationship to toxicity is important. And then also they help you define, these animal models can help you define the endpoints that you would want to pursue clinically and make the correlation to a primary clinical endpoint occasionally. So clinical development in rare diseases, as you all know, for much better than even we do at the FDA, is very, very challenging. Many of these diseases are very slow and variable in their progression, very limited patient numbers. There's a lot of information that's lacking about disease pathogenesis, particularly in the exceedingly rare diseases. And there's an absence of translational animal models in some cases. And so for these reasons, I kind of want to just touch on a little bit about what the long-term uses of these animal efficacy data are beyond first in human, because as I've said at the beginning, toxicology sort of limit the original dose, and then you kind of do some dose exploration beyond the first in human doses, but there are some specific uses for which these data are needed later on. So one of those is, very importantly, approval with a single adequate and well-controlled trial, which is how most approvals are handled in rare diseases. Those animal data can serve as mechanistic supporting evidence or confirmatory evidence. In many cases, CE is required if you only have a single study. And so these data can be very helpful in establishing that there is a treatment relationship between exposure to the drug and the outcome that was measured in the trial. So as I mentioned, they can be required depending on the magnitude of the effect observed. And together, these data with your trial plus the CE data, they form this substantial evidence of effectiveness rationale for the approval. They also can be very useful for helping to understand when it's appropriate to expose children in a clinical trial. We need to know that the children have the prospect of direct benefit. So these doses that are established and are associated with efficacy in these animal models can help us hopefully loosely guide the dose exploration in children. So we just want to have some reassurance. It doesn't have to be quantitative data, because as we all know, animals can't quantitatively predict doses in humans, but with some rationale to establish that the children have the prospect of direct benefit. So these data are used in conjunction with data from the adult and juvenile animal toxicity studies. So there are many uses for these animal data in both in the early dose setting phases and in later stage drug approvals. We really encourage sponsors to reach out at even as early as the pre-IND to discuss their plans for their first-in-human studies and how they're planning to support prospective direct benefit and confirmatory evidence, because we really want to help sponsors generate high-quality data and hopefully support the entire drug development process. Thank you.
Great. Thank you, Shauna. And then lastly, I'd like to turn to Dr. Joyce Chang, who's a statistical team leader in the Division of Biometrics 5 for one of the teams supporting the Division of Oncology 1 at the FDA. Joyce, go ahead.
Hi, thanks. Thanks for the introduction. Thanks for the opportunity to be on the panel. I'll try to just give some quick general comments. So as mentioned, I'm a statistician at FDA. And I support the Oncology Review Divisions, currently supporting Division of Oncology 1, which covers breast, gynecologic, and genitourinary cancers. But I also have some experience supporting Division of Oncology 3, which covers gastrointestinal, superficial cutaneous cancers, melanoma, and sarcoma. So I'll preface my comments by saying that my review work focuses on clinical trials from phase 1 through phase 3. So I don't have experience with animal models in the non-clinical space. And additionally, I'll note that oncology has some overlap with rare disease, since oncology includes rare cancers, but in my experience, the trials I review will generally be larger than N of 1 trials. So taking that into consideration, I do think there are lots of similarities and likely overlap in the challenges in dosage optimization that are faced by rare disease and oncology. But for now, I just want to try to summarize what we're dealing with on the oncology side, in case it's helpful. So in oncology, the Oncology Center of Excellence at FDA launched the Project Optimus initiative to reform the dosage optimization and dosage selection paradigm in oncology drug development. And a big impetus for this is because oncology drugs are changing with the shift from cytotoxic chemotherapies to more targeted therapies like small molecules and immunotherapies. Cytotoxic chemotherapies typically have steep dose-response relationships, and so traditionally dose optimization focused on identifying the maximum tolerated dose based on the idea of toxicity as a surrogate for efficacy. And so given limited patient populations and the importance of speed of development, the MTD was traditionally chosen to move forward based on few patients studied and just very limited characterization of the MTD overall. So with the shift to targeted therapies, the MTD paradigm might no longer apply, and MTD might no longer represent an optimized dosage. And the reason for that is because targeted therapies typically have different dose-response relationships that support moving away from the idea that more drug is better. And additionally, since targeted therapies are administered longer, long-term tolerability, including things like chronic symptomatic low-grade toxicities, are really important. So overall, it's important to improve from the traditional dosage selection strategy, because moving forward in drug development with a dosage that's not optimized can lead to many issues, including high proportions of dose modifications in the registration trial. Also, patients might experience poor quality of life, and that might stop them from taking an efficacious therapy due to toxicity. And as a worst case scenario, poor dosage optimization can lead to a drug not getting to market or being removed from the market. And that's why there's a push for improving dosage optimization for oncology in the pre-market setting. The FDA guidance on dosage optimization for oncology was finalized earlier this year, and it provides recommendations on this. And one main recommendation for an updated dosage selection strategy is randomized evaluation of several dosages that are within an identified dosage range. So randomization can help balance baseline characteristics across the groups of patients receiving each dosage to help with comparability and minimize bias. Of course, the challenge with the updated dosage selection strategy is in implementation and how this can be done efficiently to make best use of patient resources. From a statistical perspective, the biggest question is sample size for the randomized dosage trial, but unfortunately, there's no definitive answer here. For a trial with randomized dosages that's not intended to be pivotal, we generally rely on descriptive statistics to summarize the findings. And so the trial should be sized to allow for sufficient assessment of each dosage, but it doesn't necessarily need to be statistically powered for superiority or non-inferiority of dosages. The good news, though, is that there's a lot of research that's being done, and there's a lot of researchers developing innovative methodology to incorporate the new paradigm. The innovative methods we've seen in this regard usually focus on how to incorporate parameters beyond just safety into dosage optimization, and that includes early biomarkers for efficacy or patient-reported outcomes. However, this often involves a composite measure, and composite measures will need to be further evaluated to explore how they might correlate or relate to an endpoint of clinical benefit. So there's still a lot of work to be done, a lot of discussions to be had. That's just kind of an overview of where we're coming from and the challenges we face. So I'll stop here. Thanks.
Great. Thanks, Joyce, so much for that. And thanks to all of our presenters and panelists for presentation. I'll now ask everyone to join me on screen. I know we are running very late in time as we got a late start. We're going to go ahead and go to 12:05 for this session, so that only gives us about 7-8 minutes or so for a discussion. And so I think what I'm going to do is, I know we have the discussion questions that are listed there, and we're not going to get obviously a chance to get to all of those, but I think you've addressed a lot of these issues already. And I think I'd like to kind of focus on number 2, the discussion question, because I think this is kind of maybe the crux of the matter a little bit, in hearing some of the suggestions and ideas that all of you had, as well as all the great input and thoughts from the folks in session 1 as to what are some of the approaches. So what are kind of your key things you're seeing as approaches to dose finding activities that may compensate for the limited number of patients available for rare disease clinical trials? And I would actually add, also given a lot of the stuff that we heard in session 1, sort of the complexities and issues that patients deal with. And I'm going to kind of throw that out. And what I would like to do is kind of go around the room and have everyone kind of address that for a minute or so, because remember, we've only got about 6-7 minutes left. So I don't know who wants to kind of kick us off here.
Well,
Vikram, did you?
yeah,
Go ahead.
I sort of silently nodded, so I probably could go ahead.
Okay.
I think it's really the crux of the issue, I think, and the challenge for rare diseases, the unique piece is the sample size, and everyone's actually spoken to this. The principles of dose finding and the work that's been done in this space is, we have decades of understanding, we have decades of clinical trial design, we have new methodology and past methodology. All these things are very well established. But I think our challenge is the small size. So for me, I think a few different approaches. One, where possible, we can use a natural history registries or databases, where we can actually obviate the need for running control groups, and we can use virtual controls or controls from other databases. Intra-subject dose escalation, where possible, maximizes our information. And finally, the whole space of, which personally for me, I think is adaptive Bayesian study designs, where, you really maximize the information from each subject, you leverage your natural history database, and then you make decisions about what dose the next subject is going to get. And in that way, you try and develop an understanding of the entire spectrum of dose or concentration based relationships. And I think this is really the way to go in this particular space, not one or always equally applicable for all rare diseases. So at the end of the day, we really have to think about this for that disease, for that indication, and really understand the patients here, and then tailor these study designs accordingly. We don't sometimes have the luxury of doing multiple trials here. So sometimes, you if you're lucky, we can do healthy volunteers, but many times you're going straight into patients. So the value of each patient is so immense, and the need to maximize the information from each is hugely significant. So I think these alternate approaches are really what I think everyone has to really give a great deal of thought to.
Right.
Thanks.
And Nicholas, a minute or so for your thoughts there.
Sure. So couldn't agree more with the previous comments, but for me, the power in these studies derives not from the number of participants enrolled, but the number of measurements that are made, right? So, and of course, that bears on patient burden. You don't want to do blood draws every 5 minutes. It's not practical. It wouldn't work. But there are emerging monitoring devices. I mean, I'm wearing one, others that might have utility in establishing dose response sorts of relationships. So emerging ways of monitoring people's phenotypes could be leveraged in these trials, where again, the primary focal point is not the number of individuals, it's the number of measurements. So if those measurements are convenient and can be done in as transparent a way to the patient as possible, then one might be able to achieve appropriate statistical power by leveraging those devices. Now, having said that, those devices themselves need to be vetted. So there might need to be some work done prior to the deployment of those. But I think there's a lot of potential here. Temporary tattoos, wireless devices, imaging devices, other sorts of things that are coming online could be leveraged in these trials in the future.
Great, thanks, Nicholas. And actually, in session 3, we're going to be diving into endpoints in more detail. So I think that's great. Mark, do you have like a 30 seconds, minute for? I know it's not a lot of time, but just
no, I don't want
to.
Say any more than what's already been said here. Because certainly, in the clinic, exactly, we need to do the right measurements appropriately, so and even beforehand understand what those measurements mean. And I think again, this just simply gets back to understanding the disease. I would just also say though too, very early, in that research and early, preclinical, clinical development stage, got to understand the PK, and some animal data can help you get to that point. But I think, even as Ned and others were getting at, as long as you have some very active, interactive blood sampling, if feasible, to know if you're getting exposures that are ultimately going to provoke that intended response at the site of action, you're giving yourself the very best chance that a positive outcome is going to be seen early on in that clinical development period.
Great, thanks. And Shauna, any last second thoughts?
No, I think it's been what everyone has said has been very well articulated. I think our only point is that we are very open to discussions about flexible design. I certainly have seen a number of protocols that have implemented sort of dose titrations and other things to enable people to safely get efficacious exposures based upon what appeared to be limited toxicology that were limiting to the dose range. So, thank you.
Great. Thanks. Joyce, I'll give you 30 seconds and I'll let Vikram, he's had his hand up, just give us a final comment. So Joyce?
Sure. Yeah, I don't have too much to add. I agree with what Shauna said. Like early discussion is always good with FDA. And I would also agree with the point that I kind of add a point that I think timing of availability of data that's needed for dosage optimization is kind of a big challenge. You want to ideally have safety and efficacy signals that are reading out at similar times, but I don't think that's always possible.
Okay,
Thanks.
great. Thanks, Shirley. And Vikram, you're going to get the last word today.
Well, thank you. I had my hand up just after the comment on measurements, but I actually agree with the thing about measurements and repeated measurements that are hugely important. But the measurements by themselves don't get us to solving the question about dose response. So, I could really get a lot of measurements or meaningful measurements in a subject, but unless I do something different in terms of up titrating or down titrating or doing something else, I don't really get more information from that subject. So the challenge is, how do I maximize the information from each patient and at the same time utilize all the meaningful measurements I can get? And I think that's really the comment I want to make. And there is where we really think about creative study designs and alternate approaches as a way. And I think since the FDA is so open and receptive about this, it's really an opportunity to come in with ideas and test out what are the possibilities here, because I think that's the only way every developer of drugs and rare diseases is going to solve this problem. And I think that's the need of the hour. So thanks.
Terrific. That's a great way to wrap up the session. So thank you. And thank you to all of our presenters and panelists for participating. We'll jump right into our next session, which is session 3. And once again, I have the privilege of moderating this session entitled Integrating Sensitive Endpoints in Early Phase Trials. In this session, we'll be discussing the considerations and best practices for selecting outcome assessments and integrating sensitive endpoints into early phase trials to enhance dose finding and optimization. Once again, we have a wonderful group of experts with us, and similar to session 2, we'll have a couple of presentations followed by some opening remarks from our panelists, and then we'll open up the session to a moderated discussion. So our first presentation for this session will be from Dr. Andrew Molberg, who is Senior Vice President of Regulatory Affairs, Quality Assurance, and Quality Control at Neurogene, Inc. So Andrew, take it away.
Yeah, thank you so much, Nancy, and thank you to the organizers for allowing me to have a chance to present a perspective. I referred to it as a former FDA's director perspective on principles in rare disease, but yet as someone who is still facing the challenges of ultra rare disease drug development, particularly in gene therapy. Next slide, please. And I wanted to start the slide, which is a slide I've used for the past number of decades, on the importance of children. And you can read it yourself, but Abraham Jacobi is one of the foremost well-known pediatricians. And it really talks about the uniqueness of children as one proceeds through the drug development paradigm. Next slide, please. And I want to applaud the FDA for the recent initiation of the rare disease, hub. The mandate, specifically is calling for better integration of working together across CBER and CDER, with the goal of really improving outcomes for patients. And I particularly was trying to address the last line on intended for smaller populations, i.e. ultra rare diseases where the natural history is variable and not fully understood. My talk will be specifically on natural history, but leaving this group with a challenge, particularly my FDA colleagues, and I'll get to that in a few moments. But I really do applaud the effort of the centers to move this forward, and I wish them, and for all of us and for patients, their best fortune. Next slide, please. So the goals, as we all know, of any rare disease program are those issues that really mandate the understanding of the natural history of a rare disease. In my opinion, we've been talking about optimal doses. I'm not sure we truly can get to that, but I do think it's really important to identify an effective dose. And there's numerous examples of drugs labeled for rare diseases where the optimal dose was determined post-marketing. A good example is the treatment for Pompe disease. But underlying the decision of any relative decision making, and pointed out by several speakers in this previous sessions, are benefit risk considerations, and those need to be individualized. And that is something that I think is critical. Next slide, please. And when one considers the identification of, sensitive endpoints, when one refers to the pediatric cohorts, there's a huge difference in the developmental trajectory that impacts upon the type of outcome assessment that generally can be generated. And this graphic is well known and well worn and well discussed through ISPOR guidances and multiple FDA guidances, particularly about when the child can self-report, when it requires an observer-based assessment. And all of these are such important considerations, especially the developmental stage of a child, as it relates ultimately to the identification of putative endpoints. The next slide, please. And I think it's very clear from this previous sessions that we do need to improve our tools to understand the patient journey. Several previous speakers have commented on the challenges facing rare and ultra-rare diseases, the challenges of trying to initiate a randomized controlled trial as opposed to baseline controlled studies. And then on top of all the different patient variables, both with inpatient and between patients, their variable response to treatment, how long it takes for a patient to finally be identified with a rare disease, and then ultimately the lack of real important clinical outcome assessments and tools for assessing benefit risk. This clearly is a rare disease development paradigm that results often in a minefield without a map. Next slide, please. And it's, I think, foregone conclusion that naturalist studies are critical to deep phenotyping of rare disease. And it's particularly important in when you think about some of the multiple disease variants that exist in cystic fibrosis and Fabry disease. Batten's disease was cited earlier, plus the 9,000 others likely that have similar problems. We all understand the value of registries, and the FDA and EMA have their own definitions about how they expect these to be developed. But what's very clear is that the natural history studies that are often done are really not often the most important goal in a rare disease development program, because often the product has been already put into a test tube and therefore is post facto. And ultimately, this leads to complications and difficulties. But I would say that in order for registries and natural history studies to be effective, they must have regulatory grade evidence using consistent terminology, CDISC standards, and interoperable across the global nature of rare disease drug development. Until then, it's my opinion, these bespoke registries and natural history studies that are conducted really in their own individual ways will continue to have serious external validity problems. Next slide, please. And again, the FDA and industry understand how important it is, what's required to validate new rare disease endpoints. We've been struggling this for decades. And it's often a situation where academic investigators may disagree. But the real issue is that we have to have a change in the paradigm of developing only proprietary patient registries. I refer to that as the Tower of Babel. With that, we have lack of consistency and truly lack of coordination to expedite any rare disease program. The next slide, please. Just an old example of one that we did when I was at the FDA, working with academic collaborators, was really starting the whole entree into eosinophilic esophagitis, which at that time, back in 2012, we tried to adopt this principle of understanding the COA, approach to defining a disease with a definition, assessing natural history, and then identifying appropriate tools. I will gladly say that all that work that started back with colleagues at the FDA and external collaborators really has resulted in the approval of major therapies in this one disease. We know it can work, and there's obviously tons of examples. I just wanted to cite one from my own past. The next slide, please. So the fact is that there are many models of dose finding in inborn errors of metabolism, and they're all listed here, and we don't have time to go through, although these were points made in in parallel, potentially in other discussion topics. But there are a number of ways of dose finding that have been done successfully. And those are in the parentheses of diseases that have been labeled with those approaches. The one that I'm particularly interested in are the animal disease models that have been talked about. And I'll end my comment in a few moments just on that one specifically. Next slide, please. So the fact is, what do I see as risk to successful rare disease drug development? It's really what the hub is trying to address. The varying perspectives that may exist across the FDA centers, including level of evidence, lack of standardization, and what does seem to be an incomplete exercise of regulatory flexibility. The understanding of the natural history to target signs and symptoms relevant to a disease, and the loss of opportunities. And I will say that I would question the FDA colleagues. Can we modernize the acceptance of the animal rule, which has been used and talked about peripherally here, but in well-developed predictive animal models with limited natural history, it seems like a putative way to identify not only doses, but also efficacy that's applicable to human. Next slide, please. So therefore, I'm going to leave you with this acronym. I'll let you read it yourself because of my time limitation, but it's one that I hope that we'll try to apply in the successful rare disease drug development. My last slide is just my final slide, and it's simply to say, would you, one more, please, just to accentuate what I've devoted my career to, and that's from Janusz Korczak, who wrote How to Love a Child, and his words are ones that I live by every day that I try to make a difference in rare disease drug development. Thank you very much.
Great. Thank you so much, Andrew, for that presentation. Next, we're going to hear a presentation from Dr. JP Clancy, who is a pediatric pulmonologist and Senior Vice President of Clinical Research for the Cystic Fibrosis Foundation. JP?
Thank you so much. It's been a pleasure to listen in and be part of this, and look forward to really providing some of our experience that honestly, many of the things that Dr. Mulder highlighted, which are playing out in real time. Next slide, please. And some key takeaways that I thought, again, I wanted to just reiterate how important the communication across FDA divisions through the Innovation Hub, how important I think that it's going to be. And certainly across, but also within divisions as these interactions continue to increase, being able to ensure that it's heard throughout the different divisions, particularly when there's conversations happening with drug developers. And as we think about the second topic, choice of endpoints for phase 2 and dose selection, they can be limited. And I've certainly heard many times that the numbers of patients available for these studies to really optimize dose can be very difficult. And I think we acknowledge that there's definitely room for us to develop better, more sensitive and mechanistic biomarkers, but often may be hard for a phase 2 study to identify endpoints that really directly address how people feel, function, and survive, which would be part of their pivotal studies. I also wanted to highlight sort of the importance of considering innovative trial designs, and sort of conceptually, as been alluded to before, advancing effective versus optimal dose with the goal of optimization after drugs have sort of demonstrated pivotal proof of concept and efficacy. The importance of registries and in a broader sense, external control data sets that will really help to augment the data necessary to advance drugs for rare diseases. And then finally, something that we're running into, which I think has been alluded to too, is just beginning to really think about the challenges that come with cross-program enrollments. And I'm going to use just demonstrate some examples from our CF experience going forward. Next slide. So this is a snapshot of our CF therapeutic pipeline. And I think the point is we're very fortunate to have a number of different therapeutic targets for consideration. CF, again, is a systemic disease, and it can have targets that are very focused on treating the cause of CF, which we call CFTR based, and they're kind of on the left side of this picture here, showing different programs and different levels of either preclinical or clinical trial space. Or on the right side is really looking at very symptom-based therapies. And I guess the point is you could substitute other diseases and acknowledge that there are going to be both genetic strategies and there's going to be symptom-based strategies that could be being under advancement for the treatment and improvement of the outcomes of these different populations. But it creates a complexity because it means interaction potentially across multiple FDA divisions, as I'll show in the next slide. Next slide. And I think you can go ahead and advance, but just what we can see here is, you can advance the slide. You can see that for us in CF, we have interactions across a number of different divisions, both in CDER and CBER, depending on the nature of the therapeutic under development. And the point is that some of the conversations have very different discussions. And so you can go ahead and advance. I think we are really looking forward to this innovation hub that's really going to help to bring some clear communication across these different groups. But also we'd also need to be thinking about how we can disseminate those conversations from sort of a higher level to actually the point of contact with the partners in developing new therapeutics. Next slide. Just as a snapshot of where we are in CF, CF is really changing dramatically right now. And it's really, the reason I bring this up is because it really sort of highlights some of the common challenges that are before us in developing new therapeutics in CF. CF has been fortunate to have the development of a class of medications called CFTR modulators. And with their sort of more broad-based introduction to the CF population, we've seen an amazing improvement in median survival. But also remember that this median survival number of 61 years includes half of people who have predicted survival less and half who have predicted survival above. And as we look forward, this is looking at from 2018 up to 23, the uptake of these CFTR modulator therapies, which are kind of shaded down below, and the different therapies are demonstrated. And what we see is that there is now this subgroup of people with CF who are not benefiting from modulators, either because of their genetic defect or because they are not able to tolerate them. And this number is maybe on the order of 3,000 to 4,000 people. So really looking at an ultra-orphan disease within a broader disease state. Next slide. And what we're also coming to realize is we're very fortunate to have a number of different approaches that are really focused on bringing CFTR-based therapies to this ultra-orphan population. But when we think about drug development now in this group, we're talking roughly 1,600 adults who would be the candidates for those first in human studies who are not currently taking modulators based on eligibility criteria. Next slide. And as we get down into these populations, we come to realize that by applying some very high-level exclusion criteria, such as lower lung function as FEV1, we are already down into roughly 1,000 subjects for potential enrollment. And we know this population looks very different than what historically people think about CF. It has a much higher Hispanic ethnicity, has a much higher Black representation, and the vast majority of these people have not been in clinical trials, and really because most of those trials have been so focused on those CFTR modulators, which they have not been candidates for. Next slide. So as what we're coming to learn through our experience of trying to develop advanced genetic-based therapies or other therapies that really target that population who are not candidates for CFTR modulators, we're seeing a real change in sort of the nature of study designs, which people have alluded to. The clear delineation between a phase 1 study that looks at single and multiple advancing single and multiple doses no longer is often not able to include healthy volunteers, which again is common with a number of other rare diseases, but also puts a greater strain on the available patients to participate in these early phase studies in which they may not gain much clinical benefit. And as you can go ahead and advance the slide. As we move into phase 2, which is often, again, a very blurry line, this is really, as already alluded to, where those innovative biomarkers may take place. We're thinking a lot about how they may focus on target engagement and the evidence of bioactivity. We also appreciate that we would really hope that as we get into pivotal studies, we're able to do that with a single dose rather than have additional cohorts, which would again strain the available patient or subject population for participation in these studies. And finally, something that is unique a little bit perhaps to CF, but it may be shared with other diseases, is just the fact that when we're talking about drugs that at this point are inhaled, the ability to really have clear PK/PD information is super limited. And I think that probably shares similarities with some of these, some therapies for rare diseases, which are, while delivered systemically, may be active outside of in an organ-specific manner? How do we really sort of be able to clearly identify that dose when that access to specimens for really clearly determining that the PK-PD relationship is super limited? Next slide. So what we're thinking about in CF, which I think has been alluded to by some of the other speakers today, is sort of a very combinatorial approach for trying to really develop the data sets necessary for conducting these innovative clinical trial designs. And on one hand, we would have sort of the data that is developed by different sponsors, includes both an active cohort and possibly a small placebo cohort that contributes. And on the right side, we're seeing a variety of different external control data sets that could be utilized. One would include real-world data, such as natural history data and registries, possibly past clinical trial data, and also potentially contemporaneous control data. But all of these would need to be able to be analyzed in a regulatory compliant analysis platform. We've been working with the Critical Path Institute and the Rare Disease Cures Accelerator data and analytics platform to really essentially do this. Can we come up with a place where this data can be shared and analyzed by the appropriate sponsors and regulatory bodies to advance things? Last slide coming. Go right. Oh, you can go ahead and advance one more. So finally, a challenge that is before us that we think is really needs to be considered. And when we have a robust pipeline with a very small study enrollment pool, there is this tension between the need for long-term follow-up and how that would diminish available subjects for future studies and the interest of participants to be in future trials where benefit may be cast. And clear guidance is going to be needed to help us manage that long-term follow-up expectation against the interest of study participants to enroll in future and different programs. And so just to finalize, last slide, I just wanted to say how much we appreciate the development of the Innovation Hub. We look forward to working with you. Phase 2 endpoints, this is really where the opportunities for innovation lie. We're really going to need outside of the box thinking for our rare disease trial designs. I think many of those have been brought up today. And cross-program enrollment, it's going to be a future challenge that we have to be thinking about going forward, which is going to have impacts across a number of different disease groups. So thank you again. I appreciate this and look forward to any Q&A further down the road.
Great. Thank you so much, JP. Now we're going to hear a presentation from Dr. John Wagner. Dr. Wagner is the Chief Medical Officer at Kinexa. John, take it away.
Hi, folks. Great to be with you today. It's a real pleasure to be at a Duke-Margolis event. And my intent here is really to extend some of the comments from JP on the role of innovative biomarkers as a way of creating more sensitive endpoints in early phase studies. If you could hit the next slide. So I'm going to discuss Parkinson's disease, which, to be fair, is not exactly a rare disease. However, increasingly, many drug developers are studying narrower groups of patients, those with gene variants, because the mechanism of action of some innovative potential therapies is restricted to one of these gene variants. And so many Parkinson's drug developers are now in that same situation as rare disease, very small populations or smaller populations, which really must be optimized in order to make the best of the patients and bring the best to those patients. So that is increasingly something that's on the forefront there in Parkinson's and as you all know, across many different diseases, that gene variant approach, even as JP said in cystic fibrosis, it's narrowing a narrow population already. Next slide, please. I want to discuss the role of digital health technologies in creating more sensitive endpoints. And digital health technologies in some way sit between a biomarker and a clinical outcome assessment. Sometimes they share features of both. Sometimes digital health technologies are clearly COAs, and sometimes digital health technologies are clearly biomarkers. The it's a bit variable, but the utility of these digital health technologies remains the same nonetheless. Let me give you a quick example. Next slide. One of the activities that Connexa does is to convert features of the standard bearer endpoint in Parkinson's disease, the United Parkinson's Disease Rating Scale, into digital equivalents. And on the left is a picture of the assessment for determining whether a patient, how much tremor a patient has. And it's on a scale of 0 to 4. And as you can see, the gradations of that scale are not very fine. If one uses the sensors in a phone, you can detect on the right, you can detect tremor down to the millimeter or the sub-millimeter scale compared to that centimeter scale for the in the clinic measurements. Next slide. The application of this to dose finding is pretty obvious. In this case, I'm showing you a couple of plots, power plots of traditional in the dotted line and digital in the solid line for determining a 30% and a 50% effect size, a mimicked dose response. And what you can see is that for that lower effect size of 30%, even 1,000 patients per arm would not achieve 80% power. And in the case of a 50% treatment effect, the power is clearly much higher for a digital assessment. This is a once weekly digital assessment, and there's other ways of increasing that power further with composite endpoints and other sorts of tricks that can be used here. And you can see the obvious implication is for in a dose response experiment, the ability to really discern those doses with far fewer patients. Next slide. And digital has definitely been used already, identifying subpopulations. There's a really lovely paper in Nature Parkinson's Disease this year that shows that the power for determining a progression or the power for determining an effect is can be much better in a fast progressing population. And this was really demonstrated on the right with a Roche study where the primary endpoint failed, but when they analyzed the fast progressors, there was an ability to discern a treatment effect. Next slide. And that is replicated in our data. This is data from an ongoing study, and this is tremor assessments in the on L-DOPA and the off state. And you can see in the circled plots, there's patients that you can predetermine have very big responses on and off. And then in the right, you can see that they show actually very rapid progression in the month scale, which is quite unusual in Parkinson's patients being able to discern that. Next slide. Just finishing up, I wanted to just give you the key takeaway points. Parkinson gene variants are analogous to rare disease in this case. Digital health technologies can support more sensitive measurement versus the traditional in-clinic kinds of endpoints, really allowing for sensitive detection of progression rates and dose optimization. Last slide, just wanted to acknowledge the many people part of this work. Back over to you, Nancy.
Great. Thanks so much, John. Now we're going to turn to our fourth and final panelist to hear some opening remarks as well as some reaction to what he's already heard at this session. And that will be for Dr. Robert Shuck. He's the Deputy Director of the Division of Translational and Precision Medicine in the Office of Clinical Pharmacology at the FDA. Bob, go ahead.
Hi, thank you for the opportunity, and I'll keep my remarks brief in order to maximize the time that I can hear from and learn from my co-panelists. I just wanted to make a couple of brief points. You know, the first one I think is probably obvious at this point, and that is that the FDA recognizes that the standard approach to drug development that we commonly see used in more common diseases may not be applicable or feasible in rare diseases. But I really want to emphasize that that is as true for dose finding studies and for dose optimization as it is for any other aspect of drug development. So we often think about this more in terms of later phase registrational trials and how we can be more efficient there. But today we wanted to bring people together to talk a little bit more about earlier phase studies and how we can get better data on dose optimization for rare disease patients. So I appreciate everybody coming in and sharing their thoughts today. So the reason I think that the dose optimization is critical in rare diseases is not only because of the typical things that we think of, like, you know, you go too high and you're going to have a toxic drug, and you go too low and you might not have be getting the efficacy out of the drug. But when we have rare diseases, it's very hard to redo a study if we don't get things right. So whereas with the more common disease, we might be able to say, well, that study seemed to show a hint of efficacy, but maybe we got the dose too low. We'll try a higher dose and try it again. With rare disease studies, that's nearly impossible, if not outright impossible, to do that. So we really need to enter those larger studies with the most informed dose that we can possibly do. But we still have all those challenges that others have focused on today, such as the small patient populations and variability in phenotypes and all those things. So we want to hear from the stakeholders in this session specifically on, you know, how we can use different technologies such as digital health technologies and biomarkers to gather better data for dose finding studies and better data to inform the doses that are taken forward in rare disease drug development programs. And the last thing I'll want to point out is I think that this is very challenging for a number of reasons, some of which have already been brought up. But with dose finding studies, there's a little bit of a tension in the selection of endpoints because the clinical endpoints that are meaningful to patients and that typically support approval of a drug often aren't the most sensitive ones to detect differences in dosage. So that becomes a major challenge because we want to get data on patients that's ultimately going to support approval of a drug because we have so few patients involved in clinical trials. But those endpoints often aren't sensitive to minor changes in doses that we see when we're really trying to figure out what the optimal dose is. So I think that some of the ideas that were presented in the introductory talks, such as integrating assessments like digital health technologies, looking at innovative biomarker approaches such as omics in phase 2 studies, and using deep phenotyping approaches, may either directly or indirectly help us have more informed dose selection in rare disease drug development programs. And therefore, I look forward to continuing this conversation. And the last thing I wanted to point out is you know, it doesn't stop with the collection of the data. I think that was something that became apparent in the conversations in the last panel. It was mentioned that, you know, getting the data alone isn't going to do anything for us. We really need to think about what we do next, and that is, you know, what data do we need to validate these endpoints, and how do we use different clinical trial designs to use the information in terms of adjusting dosages or figuring out what we can do with that information and what a change in dose might mean for a patient. And I'll stop there. Thank you.
Okay, great. Thanks so much, Bob. And thanks to all of our panelists for presentations. Now we're going to move to the moderated discussion portion of this session. And I'd like to invite all of our panelists to go ahead and turn your screens back on. As a quick reminder as well to the audience, feel free to submit your questions to the Zoom Q&A function, and we will try to incorporate some of those into our discussion this afternoon. So you can see the discussion questions that we already have set up on the screen. And so I was going to go ahead and go through each of these and then sort of try to tie in some concepts that we've heard from both session 1 and session 2 here. And I invite you all to do the same. So the first one is, what are some characteristics researchers should look for when identifying dose finding study endpoints that are both salient for researchers and regulators and clinically meaningful to patients and caregivers? Obviously, we've heard a lot of the issues around the meaningfulness for patients and caregivers. And I'm not sure if salient is even necessarily the correct word here either. Maybe it's feasible, maybe it's appropriate, so I'll let you guys riff on that a little bit as well in terms of how you're answering the question. So would anybody like to sort of kick us off here?
Well, sure. I'm happy to throw myself out there. So, look forward to others jumping on. I guess I would just say that when thinking about sort of phase 2 endpoints, again, one of the primary goals, as I see it, is to really help narrow what you want to take to your pivotal studies, and with that right balance of what we think is some semblance of hopefully some measure that's pointing you towards efficacy, coupled with biomarkers that are going to be providing some level of assurance that what you may be seeing with a more clinically based outcome measure is coupled with some mechanistic mechanistic underpinnings. I guess to me, a lot of that has to do with picking biomarkers and endpoints that have reasonable precision and are going to really be able to help separate across doses. And I think the point is that it gets very difficult to get a good, clean separation in small studies, with any of these endpoints. I just think we have to acknowledge that. But I think, in my mind, I think that's a critical piece of it, and acknowledging too that it may be unrealistic to have the phase 2 or those sort of dose selecting endpoints really translate into immediately into field function and survive sort of outcome measures.
Great.
Yeah.
Go ahead, John.
I can chime in as well. I think for digital health technologies, it comes down to the application of what's practical and feasible with what patients need and want and what matters to them. And sometimes there's the best case is when there's a happy intersection of those two features. One of the other areas that we've been looking at Connexa is sickle cell disease, and the standard clinical endpoint for sickle cell disease is outcomes related to veno-occlusive events or veno-occlusive crises, and they are ratified by hospitalization. And of course, that's not something that every patient is available to all patients, especially in the global South. So we actually went out and interviewed many patients to explore some of the features there, and we found a lot of features that patients were really cared about, and the fact that many patients do need to treat those veno-occlusive crises at home is something that came in loud and clear. So again, the happy medium between what's feasible and what patients care about is usually that's the best outcome.
JP, did you have a comment to that?
Well, just as you were speaking, it just kind of reminded me that one aspect is that depending on the nature of the intervention, the nature of the endpoint that you feel is appropriate for that phase 2 study is going to vary. And I'll just give an example that we're sort of looking at. For when we're doing studies in CF in which we're doing pulmonary delivery of some sort of in a very, very broad sense, nucleic acid-based therapeutic approach that can span anywhere from ASOs to integrating viruses to editing. The sort of the value that the risk-benefit ratio sort of varies depending on which one of these, at least we're feeling, varies based on the type of intervention one's doing. In other words, the value one would get from doing a bronchoscopy for a lipid mRNA sort of CFTR replacement strategy is very different than if you had a lentiviral program that has an integrating virus, and your need to really understand what's happening at the cellular level. It's probably higher if taking that examining sort of an integrating virus or something like that, compared to something that may be very transient. And so I just don't think it's always going to be a one approach fits all strategies within sort of a general development programs.
Yeah, and I think what I've heard sort of throughout the day, and I think Bob, you brought it up as well during this session, is that you know, there's a lot of things you could measure, right? So in studies, a lot of things you can measure, and I think it might have been Danielle in the first session said, you know, I fill out all these surveys and they're not meaningful to me, and I'm wasting my time, for example. So how do you take that into consideration and weigh, because you can measure a lot of things, but you're also trying to figure out what's going to be the most important aspect, and even to narrow that further, to Bob's point, to dose finding versus, you know, broader efficacy outcomes, those sorts of things. So how do you balance that when you have such limited populations, and you know, and people with obviously dire need for new therapies? I know that's a big one to attack. Does anybody want to try to take a stab at it?
Yeah, maybe I can try to start and others can jump in. But you know, to me, the biggest things with dose finding studies is you want endpoints to be sensitive to dose, as was pointed out. Probably something that changes relatively quickly. Ideally measured in the compartment where the drug is having its effect, and very ideally something that you think is clinically meaningful as well. And that last one is often the hardest one, I think, to make the connection on. You know, that's where we get into discussions of surrogate endpoints and use of biomarkers for those purposes. But to me, a lot of that, it comes back to doing basic and translational research in terms of we identify a lot of biomarkers, and then I don't think we do a lot with them to really understand what they're telling us about the disease. I think that step is a lot harder than doing these very large screens that might you know, show you that a biomarker is up or down regulated in a certain disease. Figuring out what, you know, changes in that biomarker mean over time to a patient. And I don't necessarily mean at the level of data that we would require to call it a surrogate endpoint, but at least understanding that, you know, if you're moving this biomarker, you're doing something that's likely important in the pathophysiology of the disease. To me, maybe that strikes a balance between, you know, looking at hard clinical outcomes in these studies versus looking at, you know, a target engagement biomarker, which you know, might tell us whether the drug's hitting its target, but not necessarily whether or not it's actually going to have an important impact on the disease. So the research that gets us to that understanding, I can't tell you exactly what that is. You know, that's something that would require a bigger conversation. But I think that to me is where the ideal balance lies.
Andrew, I saw you come off mute. Did you have a comment to that?
Sure. I wanted to comment. I think for both the first two questions, I think I'll come back to my opening position, which is that unless we organize more efficient collection of data in natural history, including biomarkers, I would dare say, then we're not going to be able to refine that list, because we have one chance. And within the world of CBER, where one does phase 1, 2, and then has a pivotal trial, it's a very different paradigm with gene therapy necessarily than been done with small molecules and biologics. And that's kind of the world that I both live in now and what I lived in the past. And ultimately, the only common denominator is ensuring that you have clinically meaningful endpoints derived from patient-centric understanding. And we've heard this message already from numerous speakers who have children impacted by individual diseases. And so I'm just going to come back to that to address Dr. Shuck's comments with dose finding, but also to the issues of understanding if there is a predictive animal model, and that is the distinction that no previous speaker has commented on, then I do think that's another way in which we, for instance, have used dose targeting engagement to address clinically meaningful approaches.
Okay. And kind of maybe a little bit of a spin on this. And this is a question that came in from the audience. So what are considerations for dose de-escalation studies? Are they similar to other considerations mentioned for dose finding? So is that sort of another way to look at that? And how would that help or make things more challenging? Does anybody want to try to answer that? John?
So for a dose de-escalation study, one of the things that Robert mentioned, the linkage in the timely linkage of the response of a biomarker or an endpoint is even more important, because you wouldn't want to have a slowly responsive biomarker for a de-escalation study. So that does add an additional complication. Over to the other panelists.
JP?
Yeah, well, again, this is something we thought a lot about recently too, is escalation versus de-escalation approach. And I guess the upside of the de-escalation approach is certainly if you're not seeing effect, you kind of, you're sort of at the top end of what you think is a dose supported by your preclinical data set. You may be kind of done in some regards, or at least you can quickly move on. But I think the other opportunity it gives is it definitely gives an opportunity for at least a dose separation. If you see some evidence of activity at a highest dose, you can select a dose that's far below and definitely show a discrimination in the doses. And that may be a strategy that makes sense for ultra rare kind of conditions. So anyway, I think we're a fan of that in principle. Still working through it. I guess the last thing I was going to say is just thinking a bit about the biomarkers question is I think there's definitely a place for optimization in preclinical model systems. I think by the time you're getting into the clinical trial, you want to ensure that your biomarkers are performing in the way that you think they will. Obviously, we don't want to be putting people through potentially invasive procedures to get evidence of a tissue-specific effect with something that's not clearly reliable. And so that's just something that we've been thinking a lot about, ensuring is the risk of undergoing that sampling for us, again, usually means the lungs, could be other places, but is the risk undergoing that sampling supported by data that indicates that biomarker is really well-developed, reliable, and meaningful, as opposed to it's the first time you're seeing it off the shelf. So I'll stop.
Great. Andrew?
Yeah, Nancy, I would say that from a regulatory perspective, I don't think in an ultra rare disease, particularly one impacting pediatrics, that's even a feasible option. It's hard enough to get a first in human IND cleared in pediatrics. I would say that you're almost violating subpart D considerations. So I'd have to understand a little bit more what was meant by dose de-escalation. That's never going to be a paradigm that would be acceptable in my mind to the FDA beyond first testing a dose that is rational to study in a vulnerable population. I'm talking about pediatric subjects here.
Okay, great. And this one, this is also an audience question that I think is somewhat related. I'm not entirely sure what they mean by the phrase if safety and tolerability allows, but let me read the question. So is it fair to say that if safety tolerability allows, maximum tolerated dose or maximum administered dose is a reasonable approach to apply in rare diseases? So that might kind of get a little bit at what you were talking about, Andrew. So I don't know if you want to take a first step at that.
Well, could you just maybe rephrase or just repeat the question? Like I'm on
Sure.
Jeopardy. Would you mind repeating the question?
Sure. So is it fair to say that if safety tolerability allows, maximum tolerated dose or maximum administered dose is a reasonable approach to apply in rare disease?
Oh, okay, sure. Well, I would say that the end game, per the mandate of this auspicious meeting, is to identify, the best optimal dose. Whether that's achievable, is implicit in the investigations needing to see if you can find it. This review or person asked the question is referencing it relative to safety, not to efficacy. So again, we've all talked through this meeting about benefit risk, right? And that has to be the consideration. So it would have to be bound by appropriate efficacy considerations, not just whether it's only a safe dose.
Yeah,
Bob?
Maybe try to address each of those questions or comments. The de-escalation comment, I mean, typically we think about that with, like toxic therapies like in cancer where you need to hit that maximum tolerated dose and then perhaps de-escalate, but you really think that, inching every bit of efficacy out of that is essential. And we're really even trying to move away from that in oncology, as was pointed out in the previous session. But beyond that, there are other strategies that are kind of similar, like response guided titration is something we've thought a lot about within our office, where if there's a biomarker or a different endpoint that changes relatively quickly, you can start a patient at a lower medium dose and give them the dose that they need, as opposed to just saying, the dose in everybody's 10 milligrams unless you have a drug-drug interaction or something. So approaches like that to get to a more precise dose, I think are entirely reasonable and fairly, underutilized in drug development. I think most companies want to have a drug that's relatively simple to dose, and maybe that's more the case in common diseases than rare diseases, where we sometimes see more precise dosing because children are involved in things like that, where we can't have a one-size-fits-all approach. But certainly approaches like that, not necessarily de-escalation, but titration and other approaches can be used. The second one, the question about whether maximal dose is kind of the way to go, I would say no is kind of what I heard in the patient session. I guess it depends on what your definition of tolerable is, but we heard where even something that we might brush off as a minor side effect like sleepiness, it can have a major impact on these patients' lives. So when you get into much more toxicity than that, I would imagine that's compounded even more. So my take-home message from the first session was actually that we really do need to think about dose optimization and not just go for the highest dose possible. But as Andrew said, need to consider efficacy there as well. So,
JP?
Yeah, I just wanted to highlight one topic that may have come up in the early conversation or not, but it is just thinking about exposure to suboptimal doses and what the impact that might have on potential redosing. And again, as we think about this in certain viral vectors, we know that redosing is problematic. And so I think we just have to pay attention to that consideration that if we're choosing doses that we're quite confident or feel pretty strongly are unlikely to be beneficial, have we really done them a disservice? So I do think that has to be at least a part of the consideration as we land on these designs for dose optimization.
Andrew?
Yeah, I just wanted to say one response in reference to Robert's comments, and really referencing a previous speaker in the first session that talked about the individualized benefit risk framework that exists for families. And I think something that has yet to be done is to have a label, a USPI, that reflects not what is referred to here as the optimal dose, because I'm not sure what that means relative to interpreting his comments. But in the world I live in, I think every family with a child who's dying from a disease is going to have a different benefit risk tolerance. Some will tolerate the most risk with the best efficacy, and some will want the perfect no risk but efficacy. And I think that's missing from current labeling for rare disease. And I think this is really what the challenge I believe is important as I face the drug development and gene therapy work that I'm doing personally.
Yeah, and I do need to move on to, Bob, did you have a reaction to that?
Yeah, I was just going to say, I agree that's a very interesting concept, but there's also the concept that you wouldn't necessarily, you're not necessarily getting more efficacy by adding more toxicity. There might be a plateau in the efficacy curve, and that anything
Sure, of course,
is causing toxicity. And that means that if we're dosing at that high of dosages, that all patients are suffering toxicity that they don't need to. And that's kind of what when I think of dose optimization, particularly in this sense, that's kind of what I'm thinking is we certainly want to make sure that the drug is effective for patients, but also not having them have to suffer adverse events that they don't need to.
And I think that's a helpful nuance, because I've heard everyone sort of talk through effective dose versus optimized dose, right? So, but I'm not sure that we always are implying or meaning the exact same thing when we're talking about that, right? So because I kind of hear different things with this, and I do. So I don't know, Andrew, do you want to comment? I think you brought that up in your presentation.
Sure.
Yeah.
Sure. Again, it's my personal opinion, right? I don't have any disclaimers here except who I work for. But just thinking about my own exposure to the space, I think that the generic discussion of risk is a patient-centric factor. Patients with rare diseases will tolerate and want to have a tolerance of certain risks. And I don't think we, in the words given by a previous speaker in the morning about paternalism, I think Dr. Zinna used that word, I think we have to be careful about being overly paternalistic from the regulatory side as well. I think our responsibilities are to develop treatments that meet the needs of patients dying from disease. And that covers the effective dose, and it covers the optimal dose, and may cover anything else. And that's the challenge we haven't faced yet, at least for me.
Thank you. So I'm going to move on to question number 2 of our discussion questions here. It's what are some of the challenges, and I know some of you have already addressed this, but what are some of the challenges to selecting appropriate outcome assessments and endpoints for rare disease dose finding studies? And what are, and maybe it's more important, the latter part is, what are potential ways in which to address these challenges? And actually, I kind of want to touch on something I think, JP, that you might have mentioned, and that was challenges with enrolling in multiple studies. I don't know how that fits here, because that has been kind of a theme for today as well, in terms of, collecting data across studies to share data, those sorts of things. So I don't know if you want to kind of kick us off on this question.
Yeah, no, thank you. I guess I would just say that I think we were looking for let me put it this way, my observation and discussions with with sponsors about the concept of doing a cross enrollment is terror. In other words, they're like, last thing that we want to do is take on that risk of them having been in another study that we're now going to have sort of assigned to ours. And I think we're going to need to have a open discussion about, in fact, I think we're going to have to be a little bit proactive, hopefully from our regulatory colleagues, to be able to really help ensure that that is not a barrier. I think we know that people who are willing to participate in these studies and that could advance their therapies know that, or maybe not know, but I think they come to appreciate that the first one that comes along may not be the most effective one. And so I think that we are just going to need to be able to have a framework in which sponsors appreciate that is an important aspect of a bigger picture that they're part of. They're participating in a bigger ecosystem than just their program. And how do we sort of do that in a way that doesn't shut down we don't want the first one to the finish line to be the only one. I mean, I think we would hope that there's a path forward for future improvements, and we haven't tied up all of our patients in a variety of long-term follow-up requirements. So anyway, I've rambled. Go ahead, John. I'll pass to you.
So not related to JP's comments per se, but the broader question of endpoint selection. From my perspective in rare disease, we owe it to the patients to be able to select endpoints that are even more patient-centric than for some of the broader diseases, and in part because of dynamics of wide patient catchment areas and narrow numbers of academic or other clinical sites where these studies are being conducted. One way to really do that more patient-centric work is to do more work at home, to do more decentralized trials, to do more measurements at home, enabled by digital health technologies or not. And the question that I always raise with rare disease drug developers is why bring a patient in 200 miles to a clinical study just to do a 6-minute walk test in the clinic? It's information that can be gathered analogously at home, and those precious patient visits should be spent very carefully. Back over to you, Nancy.
Yeah, and I think, John, that ties into the third discussion question that we had too, which was how can digital health technologies provide meaningful outputs about drug response? So you outlined already in your presentation with Parkinson's disease, but do you have some additional ideas or thoughts about how to sort of move this forward?
I think that for every rare disease presents its own measurement problem, its own endpoint problem, it has to be addressed on an individual basis. There's often commonalities, like some kind of an activity measure, some kind of a performance measure, like 6-minute walk, and being able to adjust that into a setting that can be measured at home and more frequently. The more frequently is also key, because that more frequent measurements with the power of repeat measures mean more precise estimates of endpoints. So that's also very important. And I think that as I've mentioned before, one of the key aspects whenever trying to solve a measurement problem in rare disease is to put patient interviews first in determining what is most important to the patients, and what are the nuances in the disease.
Okay, terrific. So we only have a couple of minutes left. So what I wanted to do is kind of a rapid fire, just, give me your last couple of sentences, your last couple of thoughts on this whole topic of endpoint selection. And I'm going to start with JP.
I think I would highlight that the endpoints that may be selected could vary depending on the risk-benefit ratio under consideration for the drug development, and really goal would be to identify a dose that you feel confident with going forward will be both tolerated and has at least a reasonable chance of success, with a potential to compare with either self data or registry data or other external control data.
Great. Andrew?
Yeah, I think to follow on with JP, I think, we both talked about the criticality of natural history, and I would just hope that that can be standardized. And to address a comment on the question area, I would encourage FDA to really ensure that, registries and natural history collect data in a CDIS format, in standardized language, make it useful, make it global, and really try to minimize the Towers of Babel that I referred to.
Terrific. John?
Just to emphasize that digital health technologies has a real place in rare disease. It can be inherently patient centric, and often provides more sensitive measurements versus the in-clinic measurements.
Perfect. And then Bob?
I'll just say that I'm very optimistic based on these conversations that we're starting to move in the right direction. I'll reiterate something I said earlier, maybe in different words, and that is that we now have a lot of really cool ways to measure things. And I do think that validation step and figuring out what we need to do that in order to use these endpoints in meaningful ways throughout drug development, whether that be for dose finding or whether that be for registrational trials, should be a large focus of our efforts as translational scientists.
Terrific. Well, I want to thank all of you for sharing your expertise and for having a wonderful, informative presentations as well as discussion. Thank you all. And now we're going to move on to our next session of the day. So I'm going to turn things over to my colleague, Dr. Trevin Locke. There you are, Trevin. Take it away.
Thanks so much, Nancy. It's been great to hear the discussions today. Hello, everyone. My name is Trevin Locke. I'm Assistant Research Director here at Duke-Margolis as part of our real-world data and evidence work. So I'm excited to be moderating our fourth session of the day, real-world data considerations for dose finding and optimization. In this session, we'll be discussing opportunities to enhance data used for dose finding and optimization in the post-market setting by exploring the role that real-world data can play in evaluating the use of alternative dosing in the post-market setting, and by discussing some of the challenges to collecting data in the pre-market. For those of you that may be unfamiliar with the terms real-world data and real-world evidence, I'm going to quickly share the FDA definitions of them. Real-world data are data related to patient health status and or the delivery of healthcare, routinely collected from a variety of sources. Examples include electronic health records, medical claims data, data from some registries or other sources such as the DHTs. The last session was just talking about all that can inform on health status. Real-world evidence then, of course, is the evidence about the usage and potential benefits or risks of a product derived from analysis of that real-world data. With that out of the way, we have a wonderful group of panelists with us. Like the other sessions, we're going to start with some opening remarks and then have a few minutes for discussion. So to kick us off, we'll be hearing from Dr. Tina Irv, the Program Director for the Rare Disease Clinical Research Network at the NIH. Tina, take it away.
Hi, thank you for having me here today. So today I've really enjoyed participating in this meeting, and I'm glad that I've been listening all day because that's one of the benefits of going last. You get to build on what everyone else talked about earlier. So we heard, what really struck me was we heard today from the patients themselves and individuals with rare disorders about their experience. And we've heard how difficult it can be to find trials for those specific diseases. And if they do find a trial, they have to travel many, many miles to participate in the study. So we can understand why the use of real world data is tempting in a situation where patients are few and far between. And I think a lot of people look at real world data as if it's some quick fix or a magic bullet to change the face of rare disease drug development. And you always hear people talking about, we want a natural history study, we want a patient registry. Oh, you can just download our medical records and our claims data. We can gather all kinds of information about this. And But one of the big problems with this, and I'm really glad that this has come up because this is one of my pet peeves, is the quality of the data. So while this is easily accessible data, you can get to it, the quality of the data varies from registry to registry, from patient to patient, from medical records. You can have doctors across the halls who, from each other, whose medical records could never be linked or matched. So it's something that could be valuable, but I would approach it with caution. The problem is that none of that data, the real world data, is not collected specifically with the end use in mind. And I think that's one of the challenges. So they're collecting it. It's good information for what it was designed for, but it wasn't really designed for these clinical trials. We need to keep that in mind when we're using it, or how we approach it in the future, or how we suggest people develop this data that they have. So the rare disease real world data has been used in rare disease, and it has been successful, but it's also failed. And a lot of it comes down to the quality of the information that's gathered. So I work, you mentioned, with the RDCRN, the Rare Disease Clinical Research Network, which is a consortia of 20 different, or a network of 20 different consortia that study rare diseases. And the focus of this network is really clinical trial readiness. And I think one of the problems that we have is that we'll go into a clinical trial and we're looking for outcome measures, and we don't have that work done ahead of time. So I think the more work we do upfront, the better you are at identifying biomarkers, you are at looking at the data in a natural history study, and the better able you are to follow up in looking at what the patient's going to look like after treatment. So this is something I really emphasize, or our program really emphasizes. It's not the only way of doing things, but just be prepared. There are 5,000 rare diseases, and we know that they all want treatments. And while they're waiting to get into a position where a viable treatment might be available for a clinical trial, they could be spending their time with the readiness and be more prepared for what do you need to have when you come to a clinical trial. So just really getting out in front of it and really looking for data that's reliable, that's relevant. I think one of the things I really took home from listening to the parents talk, these individuals have lives beyond the rare disease. And if we collect poor data, a lot of times the clinical trial can be a life or death opportunity for the patient, and can be devastating for the family. And as they were saying, they have a lot of other things that they need to expend energy on. And by putting together poor trials or collecting poor data and having the trials fail because they're poor data or they weren't fully prepared, is a waste of their time and their energy, and is really unfair. at least I feel. So, data reliability, responsibility of collecting good data, just getting out in front of the game is the message that I'd like to really put forward as
Thanks so
my
much.
soapbox.
A good soapbox, I think. Thanks so much. Next, we're going to go to Dr. Daniel Rosenberg, the Executive Director in the Department of Global Epidemiology at Johnson & Johnson Innovative Medicine. Daniel, all yours. I cannot hear you.
Can you hear me now?
Yes.
Sorry about that. So first, thank you for the invitation from Duke-Margolis and the introduction and the opportunity to participate in this interesting and I think what will be an engaging panel with my esteemed co-panelists. I've been working in the real-world evidence or pharmacoepidemiology space for quite some time, mostly in industry settings, and with rare diseases for about 15 years now. Mostly in the area of pulmonary arterial hypertension, chronic thromboembolic pulmonary hypertension, two groupings of pulmonary hypertension, and also with certain rare genetic disorders like Gaucher type 1 disease or an ultra rare disorder such as Niemann-Pick C disease. I've been listening to some of the sessions as the day has progressed, the ones that I could participate to, and it's really brought to light the difficulty in access to data and where maybe real world data could bring some promise into the dose finding and dose optimization space. Although specifically in my experience, we haven't really used it that much in that specific space. Certainly in other parts later in clinical development and in the post-marketing phases, absolutely. But in those earlier phases of dose finding and dose optimization, I don't think there are too many examples out there. So it's great that this workshop convenes today. If I focus on NPC and Niemann-Pick C disease, where I have some experience, those who are familiar with this disease, it's a hugely heterogeneous disease in terms of when patients are diagnosed. In fact, there are 4 different groupings. If you are an infant onset for the diagnosis versus a later adult onset for the diagnosis, the disease progression is quite variable. There's variability in patient clinical characteristics. There's a lot of heterogeneity in the disease, and identifying good natural history studies, which you've heard about, or developing good natural history studies through the use of a registry, et cetera, has been a real challenge, not only to ensure that there are good natural history studies, which we did when we were developing during drug development for NPC, but did we do it early enough? Was it at the right time? Was the data available there during drug development to inform a dose finding and optimization study? I dare say not. I think we should have probably in retrospect have really tried to engage earlier in that process so that we really had a good understanding of the natural history, which I think has been a theme that we've heard at different points in time today. When I look at NPC specifically, as I said, it's a rare neurodegenerative disease, ultra rare in fact. It can present very early on to later on in adulthood, as I said. The clinical picture of NPC is extremely heterogeneous, and as I said, it starts often with visceral symptoms, and a small subset of patients with NPC die very early in life from the severe systemic disease. So quite tragic. Systemic disease, when present, it usually precedes neurological manifestations, and they are progressive and ultimately result in death. The non-specific clinical presentation of NPC often prevents a timely diagnosis, particularly for those who are diagnosed later on and adults, and patients may float around the healthcare system with a misdiagnosis. Survival is poorest in those in the rapidly progressive early infantile neurological onset, as I said, whereas patients with slowly progressive adult onset tend to live longer. So with all these complexities, if we had known more about the natural history of disease from a registry early on, would we have designed and explored a dosing differently between these different disease manifestations? That's hard to say, but I really put that out there as a potential call, a future-looking call to action to say we need this kind of information for these rare conditions as early as possible. I think about another example where I've worked in pulmonary arterial hypertension. We have developed many different drugs in this space, but I think about one in particular, which is selexipag. And this is one that you titrate up over time until it's into an individualized maximum tolerated dose. In this disease area, we have had some experience to observe in long-term post-marketing phases to see how patients actually use this. And for example, introduced a 100 microgram dose later on to allow more flexibility. And I would say some dose optimization, opportunities for dose optimization. Could we probably do more? Absolutely. But that's definitely an area to think about more. And maybe just one last word on sort of fit for purpose considerations, building on what Tina was saying in the opening remarks. This is something we consistently apply in our approach, starting with the research question through data sets, landscape scans, specific data set, qualitative and quantitative feasibility assessments, but always noting that challenge of small populations and scattered geography, whilst incorporating this notion of different underlying healthcare systems and provenance of data sets. These patients are all over the place in different geographies. That poses real problems for us to develop relevant and reliable data sets. So it really requires a lot of thought and consideration and focus to really ensure that we have the right real world data sets for these rare conditions. And I think with that, I will leave it and pass it on to the next panel member.
Thanks so much, Daniel. Next, we're going to go to Dr. Sarah Van Dries, Director of Pediatrics for the National Institute of Health's All of Us Research Program. Sarah, please.
Thank you so much. I echo the other speakers. This has just been an amazing day to learn a ton. And although you mentioned my current role, I believe my ticket to be able to attend this awesome event and share on this panel really comes from my experience before joining the All of Us Research Program. I was a physician scientist at Vanderbilt, and one of the projects that was along our trajectory in my time there was as a pediatrician and physician scientist. I used, and my team together with my team, used electronic health records and scavenged blood samples in order to do pharmacokinetic and pharmacodynamic studies in kids in an effort to understand the things that cause inter-individual differences in dose requisite to achieve clinical outcomes in kids. Specifically, some of the biggest studies we did were in opioid exposure after cardiac surgery in kids. And so the opportunities, as Tina mentioned in her opening remarks, the opportunity to glean these data and to the advantage of having something that is incredibly low burden to the patient and research participant, who here are the same in that same dual role right now, is enormous, right? The folks that we talked to about consenting to be part of our studies, when we told them, we're going to grab information from electronic health records, we're going to use leftover blood samples from what they need from clinical testing anyway, they're very excited about the prospects of what we'll be learning. Another advantage of this approach is that the data are somewhat inherently representative of what's going to happen. We focused on an inpatient settings. That's where we had really robust documentation of dosing and timing and all those kinds of things. And so our data set was the pediatric patients who are exposed to this drug in the context in which they are exposed to it, which make the data very messy. But it does, if you find the signal, it makes it more likely that it's going to generalize. But finding that signal is incredibly difficult. And again, as Tina mentioned, these data require intense scrutiny. They require intense cleaning. And three things that I learned over the time working with these data. Number one, you have to work really hard to identify and keep the real outliers while getting rid of any spurious outliers. The outliers are where the money is, right? That's where you're going to really understand differences between individuals. And that's also the easiest group to just trim off and say, well, that's not real. So you have to look really carefully at each outlier. One quick example, in the opioid data set, we had a subgroup of individuals who looked like they had incredibly high measured concentrations of drug compared to the amount of exposure they had. And we had to go back and do manual review of their entire medical record and found that they had received additional doses through bedside procedures that were documented on paper and then scanned into the electronic health record. And so they didn't show up in the flow sheets and the digitized data that we started with. So we needed to really clean that data set and understand that. That brings up a second learning, is that you need to continually repeat this process of cleaning and understanding your data set, because the processes by which the electronic health records data are even input into the electronic databases that store them change over time. And you will lose the thread of understanding what really happened to that patient if you wait till years afterwards to try to verify your data set. So this is an ongoing pursuit to make sure always that your data are matching what really happened with that individual patient. And then thirdly, the process is nuanced for different medications and different drugs. So what you learn from one opioid may be different than another, and what you learn from one drug class may be different than what you need to do for another. Again, because the documentations are different, the timings are different, the dosings are different. And so you really can't be lazy and say, well, I figured out how to do this for fentanyl, I'm just going to do the same thing for morphine. That's not how it works. You need to really develop a deep understanding of each individual data set. So given all the work that this is, and it is a tremendous amount of work, why would we do this? Number one, I think it demonstrates a commitment to learning health systems and to learning from every single patient through the experiences that they have. I think that's something we can take really seriously. Number two, I think there's been some success to this strategy. So there are things that we have learned. Number one, there's the positive findings that we've learned impact drug kinetics or dynamics. And secondly, there have been studies that have ruled out things, and I think that's equally valuable. So for example, if there are pharmacogenetic effects that we all think make sense based on in vitro models, to be able to look at those and see, and again, it may not be the definitive study that rules out any effect, but we can, with these studies, I think, rule out a clinically significant effect, and then we can move on and start looking for what is going to affect dosing for the next group of patients. And then thirdly, I think when we do this right and we do this well, we can create gold standard data sets that will fuel the methods of the future, that this can be done in a more efficient way, in a more light way. Again, it feeds into that learning healthcare system concept. With that, let me pause and pass it off to the next panelist.
Thank you so much. Appreciate the highlights on kind of how we can get to a better learning health system and learn from each patient. Next, we're going up to Dr. Klaus Romero, the Chief Executive Officer and Chief Science Officer at the Critical Path Institute. Klaus.
Yes, thank you. Somehow my video is not responding, but okay, let's go with that. So yeah, thanks for the invite, and really enjoying the conversation. I'll bring the perspective of what we've done to tangibly integrate patient level data from clinical trials, observational studies, and real world data sources together, not for the sake of integrating data, but for the sake of leveraging those data to turn those data into actionable solutions that can accelerate drug development. And to illustrate that point, I'll use an example in Friedreich's ataxia. So there was a question in the chat that I saw. I'll let the agency comment on their specific plans. I'm not going to speak for the agency, but I'll share our perspective. When we started integrating data from clinical trials, of course, CDISC was the way to standardize the data. Great. CDISC was developed for that purpose. Once we started integrating non-clinical trial data sources, observational patient registries, health records, that no longer worked. And so what has been our solution to that? Ontologies. And so OMAP lends itself really well to take unstructured data and structured data and be able to integrate them. And from there, build analysis subsets that you can put in CDISC compliant form to be presented to regulatory agencies. So that's one aspect of that journey. We have been able to integrate 700,000 individual records across clinical trials from industry, observational studies from academia, patient registries from patient groups, and electronic health records. We have the largest integrated database in Friedreich's ataxia, which is, as you know, a rare disease. And we have, thanks to FARA, the Friedreich's Ataxia Research Alliance, they partner with us to put all those data into an integrated platform that FDA was visionary enough to fund starting in 2019. And what that allows us to do is to have a data analytics platform that looks really hard and enables the diagnosis of data quality, data reliability, and for specific purposes, because not every single question will have the same answer regarding if the data is reliable, the quality is there, and the information value is to be met depending on the question you want to answer. What happened in Friedreich's ataxia? So at the time, the company, which is now acquired by a different company, had a successful phase 3 trial that had met the statistical endpoint. There was a statistically significant difference between the groups. Okay, how many times have we seen that, leading to the question, does this statistical evidence constitute actual evidence of benefit for the purposes of meeting a regulatory decision? That was exactly the question in the case of the Friedreich's ataxia example. The trial had met the statistical requirement, but the question was, does this really constitute evidence of benefit? In the classic sense, the answer would have been, hey, sponsor, run another trial. Thanks to the fact that the agency had funded the establishment of the data analytics platform and that FARA had shared significant amounts of data with us, and we were able to integrate those data into the platform using the ontology setup, we had the resource for the follow-up, which was, can the company go into the platform and run a propensity matching score analysis to extract external controls that could be part of the total evidentiary package? That's exactly what they did, knowing that all the work to integrate the data, diagnose the quality of the data, diagnose the reliability of the data, the standardization of the data, the integration of the data into the platform was done, and that there was an interface for the company to be able to run that analysis. The company did just that. And that shaved off a significant amount of time. And the company was able to put that together as part of the total evidentiary package for the agency. The agency has full access to the platform as well. So the replication of what was done was really easy to do. And that led to the conclusion that with the addition of the external controls, the augmentation of those data, the evidence of benefit could be demonstrated. And that led to the first approval of the first ever disease-modifying treatment for Friedreich's ataxia. What's new? External controls have been around for quite some time. The agency is very receptive to that, as long as, of course, the question is fully understood. The data can actually support the generation of those external controls. There's guidance documents on that, so there's no question about the willingness and the receptivity of the agency to do that. But certain case-by-case requirements need to be met. So that has been around for quite some time. Data standardization has been around for quite some time, but the ability to integrate data from multiple sources in a way that allows the generation of those analysis subsets for the purposes of supporting regulatory submissions is added value. And what this all did was it really improved the efficiency. So something that could have taken, after the initial interaction between the sponsor and the agency, could have taken 18 months, 24 months or more, took way less than that. And that led to a pre-pivotal approval in a pretty devastating disease. The platform is there. We have, like I said, 700,000 individual records across 30 plus indications, thanks to the contributions from industry, academicians, patient groups, and health systems globally. We have data from the US, we have data from Japan, we have data from Europe. And it's there for a reason, to support the acceleration of drug development. So just wanted to give that tangible example that things can be done. There are examples where the data were diagnosed as not being quite ready for prime time, but that's okay because it's in the system loop. Us being able to communicate back to groups and the people collecting and designing the collection of the data to optimize their data strategy so that the data can reach the same level that the Friedreich's ataxia data reached. And so the future is now, and things are happening, and it's a really exciting time. I'll leave it at that for now. Thanks, Trevor.
Thanks so much, Klaus. Fantastic example to help kick us off. Next, we're going to go to Dr. Raj Matabushi, Associate Director of Guidance and Scientific Policy in the Office of Clinical Pharmacology, and the Director of the CEDAR's Quantitative Medicine Center of Excellence at the FDA. Raj, all yours.
Thank you, Trevor. Am I coming through clearly?
Yes, you are.
Thank you very much. And thank you to the Duke-Margolis for inviting me to participate in this excellent workshop and also as part of this panel. And as it pertains to real-world data considerations for dose optimization and dosing related, my view or my approach to this would be a tad bit different from what my fellow panelists have mentioned so far. They have provided real good insights on the challenges with the information or what kind of standards that needs to be there. My approach would be primarily from a quantitative medicine kind of a perspective, where if we have good real world data, then it can be very powerful, and the promise of it is what I'm going to touch upon. So fundamentally, I believe dosing optimization is an ongoing exercise for any drug or any drug development, not just in the rare disease space. It's generally the case across all the therapies that are out there. The fundamental reason being our pre-market efforts are primarily, as much as we try to get the right dose, there is always a balance, a pragmatic balance that is hit, as several of the previous speakers in other sessions also pointed out, that we are looking for a safe and effective dose. So sometimes we do get we do achieve a reasonably good optimization of the dosing, sometimes not, but we would have met a regulatory standard with respect to benefit risk. So the dosing optimization is an ongoing question that always happens, and it has and this information especially becomes important from a real world perspective, because that is where the questions around what aspects of dosing do need to be optimized or altered becomes a question. And we have seen this for drugs for chronic diseases, where there have been efforts where, for example, we have had one example for cetuximab, which was previously approved for squamous cell carcinoma, for head and neck cancers and colorectal cancer, where it was approved for weekly regimen. And based on some elegant modeling and simulation work that was done with available data as well as real world data, they were able to show that even a twice weekly dosing regimen could be similar in its benefit compared to what was previously studied and approved. And this was a very important question to be answered, especially during the COVID-19 pandemic, where where it had the potential to decrease the time spent at the clinics, which was of importance. So there are going to be a variety of questions that do come up, especially when treatments are approved for lifelong use. I think fundamentally there is an opportunity, if the right kind of data exists. I think using quantitative approaches could be very complementary to leverage the learnings from such information and translate them to inform the dosing or optimize the dosing. Though I have not worked significantly in the rare disease area, one other example that comes to mind is the enzyme replacement therapy, the first generation enzyme replacement therapy, Lumizyme for Pompe's disease, which was approved at 20 mg per kg every 2 weeks as an IV infusion. This was a significant treatment for the condition. And once it was approved, based on the real world data, information started emerging that that there was more room for improvement with potentially higher doses, and maybe there are subsets of populations such as those with infantile onset Pompe disease might actually benefit from greater doses, higher doses. And in fact, there were studies with 40 mg per kg once every 2 weeks that were studied. And in fact, some of the findings also indicated maybe early treatment and more frequent treatment could also be, or more frequent dosing could also be useful. So there are signals and information that do come up, and sometimes they could be very useful in informing the next generation therapeutics that do come up, because one of the thought process was higher doses might actually result in a greater uptake to the target sites where the concentrations need to be there, which can help the next generation therapeutics to actually target those which might have a greater uptake or may have a longer shelf life of the enzyme activity for the enzyme replacement therapies. So all in all, I think there is a sweet spot here that can be hit. There is a lot of knowledge that comes from real world data, and that can definitely inform various dosing related questions.
Thanks so much, Raj. I'll ask all of our panelists to come back on a screen. We've got just a few minutes for discussion. I think in those opening remarks, we both heard some examples of what's possible when things work, but also many of the challenges that we might face to getting to that point. I'll send a quick reminder to the audience that we'd love to have your questions through the Zoom Q&A feature. We don't have a ton of time for discussion in this section, so get them in now, and we'll try to get to them if we can. But for now, we've got a few prepped, so we'll try to get through a few of those. Daniel, I may go to you first. We talked a little bit about the challenges of collecting data in small populations, and perhaps the role of rural data in helping address some of those challenges and being an opportunity to expand what we know there. But can you talk a little bit more about some challenges that we might have in collecting rural data in rare diseases, and how we might think about solving some of those? You mentioned kind of the small populations across the globe. So how can we leverage rural data to help with that?
I mean, I think traditionally registries or sort of longitudinal observational cohort studies have been kind of the go-to, particularly for rare or ultra rare conditions where you may have one patient here, one patient there, one patient here, and really scattered around healthcare systems and scattered around the globe. I think some of the other panelists have mentioned that with increasing technology and ability to perhaps use EHR data in a more efficient manner that can either connect EHR and then with some additional data collection that's relevant to your rare disease. Often, for even dosing information may not be readily available in some secondary data sources, so you may need some primary data collection, a mix of the two. I think with increasing technology, digital tools, allowing EHR data to go directly into a data set that you augment with primary data collection, more and more is possible. I think one of the challenges in rare conditions, and is a theme I've heard a lot, is on the one hand, we say, well, let's go out and collect all this information and then we'll use it for lots of different purposes. Of course, the best data set is the one where it is fit for a purpose and you've designed it completely properly. So on the one hand, you say, well, which one do you do? If you do something in the middle, maybe it doesn't satisfy any of the questions you want to do. So I think that's a balance that one really has to think about carefully when designing a study for which you are collecting data versus just collecting data. I'm a fan of the former, and that you have a really clear research question and purpose, and you collect data specific to it, or you assess secondary data to see will that fit your needs. That said, in terms of potential solutions, we started to work a lot more with federated health data networks, and really for rare disease specifically, allowing data to sit behind firewalls and sit in different academic centers, and getting large enough populations. That's another possibility to explore. I mean, there are different solutions out there, but it really requires, you know, clarity of your question, efficient and appropriate understanding of what kind of data you want to collect so that it's really going to answer your question. And if you don't have that data available, well, then augment it with some primary data collection, so that you have the right data set that has little missingness, it's appropriate, it's relevant. I really want to emphasize it's relevant, it has the right information in it. And of course, it's reliable, it's good data quality, not reasonable, but good data quality that will meet a regulatory standard, etc.
Thanks so much. Does anyone else want to weigh in that? I saw some head nods and reactions from other panelists.
I
No?
thought it was really, it was a great statement, Daniel. Right on target.
Okay, so not seeing any from the audience just yet, so I'm going to keep working through our list that we started with. Sarah, I think you noted some of the work that you did in your prior role around combining EHR data sets with some samples to get to PD information. I think that's often lacking in normal world data sets. Any kind of additional lessons from that work on how we might leverage world data sets effectively to kind of get at optimal dosing of drugs, whether we have samples or to pair with it or not?
Right. So I think you know, we started with the pharmacokinetic first, and then layering on the pharmacodynamics after that approach, which led itself to needing samples to understand some of the drug concentration variability. If you have you know, depending on the profile of your drug and what you're looking at, again, if the PK is very predictable and stable and not so variable in individuals, or has very known inputs, you might be able to go to a PD first, or a combined PK/PD study that may be less dependent on grabbing samples and knowing concentrations, all those kinds of things. A key issue to that, and I think we've heard about it already today, is having the right endpoints and knowing what you're going after. So getting input early on, and I think this also kind of builds on some of the prior comments about maybe the clinical trial isn't there yet. What should the advocacy community, what should the patient community, what should the research community do if we're not there yet? Well, figuring out what the right endpoints are, what are we trying to achieve with a therapeutic, is only going to help us get in that right direction, because we need to understand the variability of that endpoint amongst people who are not yet treated. We need to understand how to capture it reliably. We need to understand how it changes over time. So all of those things are so very important, and there's no way to move forward without having reliable metrics. I think that's a theme that you're hearing all of us say.
No, absolutely. And actually, I think that dovetails nicely into a question that just came in from the audience. How can we think about optimizing study design, trial design, recognizing that the data is not always ideal? Kind of how do we think about kind of in the short term, as we're figuring out kind of the right endpoints and everything that you just highlighted, Sarah? Like, how do we get a drug out that's in the best interest of patients with kind of maybe not the best data, while we try to figure out how to get to that better data you were describing? Does anyone want to try to tackle that, Klaus?
Yeah, that's a fantastic question. That's a question I would grapple with all the time, because even though clinical trials, a lot of fantastic attributes that other types of data sources don't have, other data sources have attributes that clinical trial data don't have. Longer term of follow-up, inclusion of additional sources of variability, which is a good thing as you try to identify those covariates and those sources of variability and quantify uncertainty and that kind of stuff. I don't know that I like the notion of the waiting for the ideal data. And especially in rare diseases, every data point is precious. And there are ways in which we can extract the maximum value of those data sources. And it links to proper data integration before you start having the urgency to answer those questions. And like in the previous example, start integrating the data, make sure that the data are curated, because when the questions come, if you have data that are outside of a clinical trial, but data that can support the answer to very specific questions in drug development, that's when you gain the efficiency. The modeling examples that Raj was indexing on, making sure that you not only integrate the data in advance, but you start quantifying and modeling disease progression. What are the sources of variability? What are the difference in trajectories? Those types of things help a lot when the urgent questions come. And I'll leave it at that.
Thanks, Klaus. Anyone else want to jump in on that one?
I was just going to add, having the data have its provenance, so you have those questions. It may not be exactly what you want, but you have a good understanding of the quality of that information, and so you can trust not exact information that is very understandable. It has great metadata. You really have an understanding of what it asked and how it was answered. That can be valuable as opposed to the exact fit. It's a good fit, not the perfect fit, but a good fit and a reliable fit.
Sarah.
I'd just add that I think to the degree possible, understand exactly where the data doesn't fit, right? And then there are ways that you can model the worst case scenario. How wrong are your data and how does that change your results? There are times when you work that out and it turns out that there is some, your findings are robust and you can move forward. There are times when you need to get a new data set because your findings are absolutely not robust and you can't trust it. I would, as a pediatrician, we have a saying, never trust a baby. I would also never trust a not collected for purpose data set. Even a fit for purpose data set, you're still going to test it and make sure it's right? Never trust a data set. You have to test it, prove it, make sure it works, and then know what its weaknesses are. It's not if it has weaknesses, it's what are the weaknesses, and then see worst case scenario, how much that changes your outputs.
Thanks. Raj, you had your hand up. Did you want to weigh in?
Yeah, I think between Klaus and Sarah, I think they put it very elegantly here. I think what is done in the clinical trial and what happens in the real world, there are two different objectives with the data, two different types of flavors of information you're getting. And one piece I want to say is using quantitative methods in a very targeted manner can actually help integrate the learnings across these two and help maximize that particular information to inform various questions, not just dosing, I would think.
Thank you all. We're nearing the end of our time. Maybe as a closing question, knowing that our last session of the day is focused explicitly on pediatric populations, I'll ask if there are any thoughts from any of you on how we might leverage world data sets to kind of follow patients over long periods of time that might be taking, a drug for rare disease over a long term. Is any additional thoughts on how we leverage real data sets to kind of have that long-term follow-up of how patients might respond and how that might impact, how we think about dosing if a drug is needed over a long term? Sarah.
I promise this wasn't a spike from the audience. This does give me the opportunity to talk about my current role in the All of Us research program. This is the idea of this program is that is much longer longitudinally than what would be supported by a typical NIH grant, something that is able to follow people for decades to come. And I think all of us and other programs like this that are funded not on the typical cycle of an individual investigator-initiated project are what are necessary to be able to follow folks over the very long haul.
All right, thank you. I think we are at time. If no one else wants to weigh in, I believe we're going to a break for about 15 minutes. or just shy of. If everyone can please be back for the next session at around 2:20 Eastern. Thank you all. And thanks to our panelists for a great discussion.
Welcome back from our break. Thanks for joining us for our last session of the day. This one's titled Defining and Optimizing Doses for Pediatric Populations. My name is Garrett Homry. I'm Research Director for Biomedical Regulatory Policy at Duke-Margolis, focusing primarily on our FDA portfolio of projects. It really is a pleasure to serve as your moderator for today's 5th and final session. Couldn't be more grateful for the folks that are about to join. In this session, we'll be listening to a case study presentation specific to pediatric rare diseases, and then opening up to a panel discussion to better understand some of the special considerations for dose finding in these kinds of populations, also exploring some next steps for applying these approaches. We have a great group of experts with us today. I'm first going to hand it over to Dr. Rojing Li, a clinical pharmacologist and team leader in the Office of Clinical Pharmacology at FDA, to kick us off with a case study. Rojing, whenever you're ready.
Okay, thank you so much. Can you hear me okay?
Perfectly.
All right. Okay, thank you for the opportunity. And in this session, I'm going to use a case example to illustrate how pediatric extrapolation is going to be used in a regulatory decision-making for pediatric rare disease. Next, please. So this is just the disclaimer. And next, please. So as we all talked about in the previous 4 sessions, the drug development for pediatric rare disease is very unique and challenging. And nearly all drug development programs for pediatric rare disease have to deal with the very limited number of patients. And this makes it very challenging to enroll the youngest age group of the target patient population in the clinical trial. Next, please. Additionally, we often see the disease heterogeneity in the pediatric rare disease. For example, the earlier onset genetic conditions may progress more rapidly than the adolescent or adult presentations. And also, we see different genetic variants exist in the same disease, which may cause the difference in the treatment response in terms of both efficacy and safety. And another point is that due to the natural history of the disease, the same clinical efficacy endpoints sometimes cannot be used across the whole age spectrum. For example, efficacy endpoints may be appropriate in adolescents or adults, but it may not be interpretable in younger age group, which actually makes the efficacy evaluation more difficult. Next, please. In addition, the difference in PK introduced by the administration issues, body compositions, metabolism, and excretion difference, as well as feeding issues in young age pediatric patients compared to adolescents or adults, also places additional challenges. Next. Furthermore, if you want to initiate a pediatric clinical trials for drug development, you will also need to keep in mind of those ethical considerations. For example, here I put prospects of direct benefits based on evidence to support the proof of concepts. So the last bullet points, we have been talking about it a lot today already, and I would say it's probably one of the most difficult challenges that we have to deal with in the drug development for pediatric rare disease is the large knowledge gap in the natural history of the disease, which make it very hard to identify what appropriate clinical endpoints or efficacy assessment tools should be used to evaluate the treatment effect. And as you can see in these 4 blocks, there are many challenges in pediatric rare disease drug development. And in my presentation today, I want to focus on one approach that can help to deal with the challenge of limited number of patients. And this approach is pediatric extrapolation. Next, please. So as I mentioned earlier, the small number of patients within one rare disease and their wide geographical distribution make it very challenging to enroll sufficient patients in a clinical trial. However, for rare disease affecting both adults and pediatrics, when data are available in adults, extrapolating adult efficacy to pediatric patients may be feasible if three conditions can be met. The first, the course of disease progression is the same in adults and pediatrics. And second, the pharmacology of the drug is similar in adults and pediatrics. And third, the response to the treatments is anticipated to be similar. And if above conditions are met, the most common strategy used in pediatric extrapolation is exposure matching. Here, the reference exposure used in the exposure matching for pediatric dose selection is usually, in general, the exposure in adults associated with an established efficacy and safety. Next, please. So today, I will use nexviazyme as a case example to illustrate how pediatric extrapolation could be utilized in dose selection for pediatric patients to treat Pompe disease. And the Pompe disease is an inherited disorder of glycogen metabolism caused by deficiency of the lysosomal enzyme, acid alpha glucosidase. That leads to the glycogen accumulation in skeletal and cardiac disease, muscle, causing muscle weakness, respiratory failure, and heart failure. The incidence of Pompe disease is about 1 in 40,000 births. And nexviazyme is a hydraulic lysosomal glycogen-specific enzyme indicated for the treatment of patients 1 year of age and above with late-onset Pompe disease. In the clinical trials of late-onset Pompe disease of this drug development, no patients younger than 16 years old were enrolled, while the approval decision was made down to 1 year old. So the approval decision for pediatric patients with late-onset Pompe disease weighing less than 30 kilograms was based on the pediatric extrapolation. Next, please. As I mentioned earlier, to use the pediatric extrapolation, the three conditions of similarities need to be met. They are disease similarity, pharmacology similarity, and response to treatment similarity. And in this specific case, the pathophysiology across age spectrum of patients within the late-onset Pompe disease is similar. And the mechanism of action of nexviazyme is enzyme replacement therapy, which is the same in adults and pediatrics. In addition, the response to nexviazyme was expected to be similar in adults and pediatrics patients as well. So conditions of these three similarities between adults and pediatrics were met, which provides the foundation to conduct pediatric extrapolation. So the next question would be what clinical and scientific data we had to inform the dose for pediatric patients. As shown in these slides, in the middle light blue box, the first bullet point, the efficacy of the treatment, was established in adult patients with late-onset Pompe disease in an adequate and well-controlled trial, which is listed in the block at the left side. And in this trial, the study dose was 20 milligrams per kilo every other week in patients with late-onset Pompe disease, 16 years old and above. And this trial also provided PK data for the population PK analysis. So the exposure associated with this dose in adult patients was the target exposure that the pediatric extrapolation would need to match. In addition, the applicants also conducted an open-label safety trial in pediatric patients with infantile onset Pompe disease down to 1 year old, which is shown in the block at the right side. In this trial, 20 milligrams per kilo and 40 milligrams per kilo every other week dosing regimens were studied. And this trial provided PK data and safety data from pediatric patients down to 1 year old. So with above efficacy, safety, and PK information, the dose for pediatric patients with late onset Pompe disease was selected based on exposure matching using population PK modeling and a simulation approach. Next slide, please. And this plot shows the simulated exposure in different weight groups following 2 different dosing scenarios. In the 20 milligrams per kilo every other week, is shown in the pink boxes, and the 40 milligram per kilo every other week is shown in the blue boxes. And the x-axis shows the different body weight groups, and the y-axis is the drug exposure. And there are also a few dashed lines in the plots to mark some important exposure range that we wanted to match. For example, the 3 blue dashed lines from top to bottom mark the 95th percentile, median, and 5th percentile of the exposure in adult patients from the adequate well-controlled trial, which is also the target exposure. And the green line at the bottom is the minimum exposure from this adequate and well-controlled trial. And the red line on the top is the maximum exposure in the pediatric patients with infantile onset Pompe disease, who received 40 milligram per kilo. So we consider this as the safety limit. The dose selection for pediatric patients with the late onset Pompe disease was based on matching the simulated exposure to the target exposure. So the approved dosing regimen is 40 milligram per kilo for patients weighing less than 30 kilogram, which is shown in the blue boxes at the left side of the black vertical line in the plots. And we approved 20 milligram per kilo every other week for patients weighing 30 kilogram and above, which is shown in the pink boxes at the right side of the black line. Next slide, please. So pediatric extrapolation has been widely applied in regulatory decision-making. However, there are still a lot of residual uncertainties and knowledge gaps that need additional efforts. For example, when it comes to the exposure prediction in infants and neonates, the maturation of the metabolism enzymes as well as organs can introduce more uncertainties in drug exposure, especially for small molecules. And in some conditions, mechanistic-based modeling approach, such as PBPK model, may be used to help answer more complicated questions. Next slide. In addition, this is often the case that safety information is not adequate to justify the dose in a young age population, which made the dose selection for this group based on extrapolation very challenging. Next. And furthermore, the knowledge gaps, as I mentioned earlier, in the disease heterogeneity and the natural history introduces significant uncertainties in ethics and safety evaluation, making it extremely difficult for pediatric dose selection. And I would say it definitely needs more research and explorations. Next. Next. Oh, sorry. So acknowledgement, please. Next slide. Yeah, so this brings me to the end of my presentation today, and I want to thank the review team for the next design, as well as my clinical pharmacology review team at DTPM. Also, I appreciate the team from Duke-Margolis for having me in this session, and thank you so much. Now I'll hand back to Gary to continue our session. Thank you.
Rojing, thank you so much, both for talking about the unique challenges associated with dose finding in pediatric populations, but the ability to tie it to pediatric extrapolation and share an opportunity of a tool that has actually been in use and can hopefully be applied elsewhere. Next up, brief opening remarks from the rest of our panelists. Before we go to open discussion, let me first start with Dr. Thomas Miller, Vice President and Global Head of Acute, Chronic, and Pediatric Disease Nucleus in Bayer's Pharmaceutical Division. Thomas, please.
Thanks, Garrett. And can I get some technical help turning the camera on? I'm having some difficulty doing it. Oh, there we go. There we go. Okay. So thank you, Garrett, and the rest of the Duke-Margolis team for the invitation today to be able to share some thoughts regarding one of my favorite topics. As Garrett indicated, I have a role overseeing a development portfolio at Bayer, and a portion of that has been near and dear to my heart for a very long time has been our pediatric development initiatives. So these are topics that our pediatric development team has to think through on a day in and a day out basis. I think Dr. Lee gave a great talk to open the session up, and what I thought might be helpful was to highlight a couple of the concepts that she raised in the context of a sponsor's perspective. So maybe starting off with the topic of ethical considerations. Of course, children do not have the ability to advocate for themselves, so it's the parent or the caretaker's responsibility to provide informed consent for the child's participation in studies. And as she alluded to, studies that are solely focused on understanding pharmacokinetic dynamics may not have appeal to the parents, and may raise some questions with IRBs from institution to institution. So one solution that we've employed on this on this front to ensure that we can collect the data that is necessary, but also maybe broaden the definition of benefit risk for participation in the trial, is to think through a design that might be adaptive, for example, where you may begin with a PK optimization phase for extrapolation and follow with subsequent assessment of safety, or perhaps even just considering a long-term follow-up where everybody that participates ultimately has the opportunity to continue on with the study at the optimized or selected dose. The second topic that I thought was one that is quite relevant and that teams like mine spend a lot of time thinking about is the exposure uncertainty. And very specifically the metabolic fate of the drug as the child ages, and this is particularly challenging for chronic diseases that emerge early in life, perhaps as early as the neonatal period, because the metabolic fate of any given drug may be different for a neonate versus an infant versus a child and versus an adolescent. So there are many discrete cohorts throughout the childhood period where this needs to be considered in certain circumstances. And finally, I think Dr. Lee gave a very timely example regarding the enzyme replacement therapy and Pompe disease utilization, because this is a field that has become, that has reemerged as one that's of interest for the life science ecosystem, specifically relating to genetic medicines, gene therapies, and cell-based therapies. And I think many of these concepts that Dr. Lee very nicely summarized for us will need to be revisited because in certain diseases that persist into adulthood, like late onset Pompe disease, we may have the ability to start with adults and work our way backwards as appropriate and where risk benefit allows. However, for other types of Pompe, most notably infant onset, this might provide our ecosystem with a bit more challenge to think through how to best handicap dose. So hopefully a bit more time on this topic when we move into the discussion. So with that, I will hand over to Bryce Reeve from Duke.
Bryce, up next. Thank you so much. Thomas, appreciate all those points. Look forward to getting back to some of them.
Thank you very much. And thank you for allowing me to be part of this really important panel. I'm Bryce Reeve. I'm a professor at Duke University School of Medicine and director for the Center for Health Measurement. Our center both leads and facilitates the design and use of clinical outcome assessments in clinical research trials and other types of studies. And so, given that context of the field I work in with clinical outcome assessments and thinking about this particular conference or meeting, when thinking about trials there and what their goals are, so we do know, and everyone knows, that in these early phase trials, the goal is to help identify what is the optimal dose level And we know that optimal dose level in early phase trial depends greatly on the patients receiving the drug being able to tolerate the drug under study. In later phase trials, phase 2 and phase 3 and so on then the goal is to identify effective treatments. And we know that identifying effective treatments depends on the evidence that there is a treatment benefit. For treatment benefit, the FDA defines benefit as how a person feels, functions, and survives. And so for either of these goals for early phase or late phase trials, the patient's perspective is critical, including pediatric rare disease studies. So ideally, if we are trying to collect information on tolerability and benefit, that this comes directly from the patients themselves. It's recognized that the patients are the gold standard for how they feel and function. It's a very subjective experience. Thus, to the extent possible, the patient should be included in determining what symptoms, functioning, and related health and quality of life domains are included in the assessment, and to the greatest possible extent, the assessments we use, the clinical outcome assessments, should directly capture their voices. In the pediatric research field, it is commonly accepted that children as young as 8 years of age can independently read and respond to surveys. And there's quite a number of health-related quality of life surveys out there for children as young as 8 years to complete. However, when the child may be too young, too ill, not developmentally able, or just simply doesn't want to complete surveys, then we have to start looking to other sources to collect this data, including using other reporters, such as parents or clinicians. However, when we do include these alternative reporters, we are introducing more bias into the assessment of how the child feels or functions. There are a number of research studies that have looked at the congruence between caregivers and clinicians with the child there, and the evidence supports that the family caregiver, the parent, typically knows their child the best. And so where that leaves us is to address these key goals of these research studies. We have these key questions that we must address. Are we measuring the right domains to assess treatment tolerability and benefit? To what extent in assessing and determining which domains we use are incorporating the child's voices? Number 3, do we have the right regulatory grade measures for the child or the observer to complete to assess these symptoms and functioning? And then importantly, number 4 is, how can we use these clinical outcome assessments and implement them in clinical trials to address these study goals, including how we use the patient information or the caregiver or the clinician information to help inform drug tolerability assessment? If there are additional time in this panel session, I'd love the opportunity to talk about some of the measures that we've been developing for newborn babies up to older pediatric patients to look at drug tolerability. Thank you.
Thank you so much, Dr. Reeve. Capturing clinical outcome assessments and the identification of treatment benefits informed by direct patient feedback and the challenges associated when you can't get that direct feedback. So look forward to hearing more about specific examples of measurements when you're in that really difficult scenario. Up next, Dr. Alexander Vinks, partner at NDA Partners. Sander, please. I can't hear you. Did you come through that time, folks? Try again, Alexander.
Yeah, I can't hear either.
Try once more, Sander.
Maybe Sander can come in by phone.
Well, how about let's do this while he's sorting out. Gil, what do you say we jump to you and then we'll try again with Sander after? Our last was going to be Dr. Gilbert Urquhart, Associate Director for Pediatrics, Office of Clinical Pharmacology at the FDA. Gil, what do you say? You jump in, and then we'll get Sandra on the back end. We'll figure this out.
Okay, thank you. It says the host has stopped my video.
Hmm.
Is that
Can I get
you?
somebody on my end to help with
Start
that?
my video.
There we go.
There we are. Okay, great.
And Sandra,
I'm
we
Bill
will get to
Burkhart.
just.
I'm associate. Yeah. I'm Associate Director for Pediatrics in the Office of Clinical Pharmacology at FDA. And I was a faculty member researcher at 4 major children's hospitals before I came to the FDA. Been a great discussion today. Thank you very much. And I've got a lot of notes, but I picked out 3 things that I'd like to mention. One about study design, one about dose ranging, and one about long-term safety assessment. The first is the N equals 1. I know that always scares people. Assam mentioned that right at the beginning. People don't know what that is exactly, and so I would encourage you to kind of go back if you're not familiar with N equals 1. It's been around for 70 years. I would encourage you to kind of go back and read about it. We had a program about 10 years ago where we had 4 different products that were submitted for pediatric neurogenic bladder, and we only approved one of them. That one was done with N equals 1 type of design where the patient were their own control and where you could actually show that there was, for individual patients, there was an effect. And so if you want to, it's in the Journal of Clinical Pharmacology 10 years ago, 2014. But that is a design, particularly for small patient populations, and in pediatrics, it seems like they're all small patient populations, that I would encourage you to go back and look at. The second thing is about dose ranging. And we've just recently reviewed dose ranging. You know, the three major types of dose ranging are, of course, parallel, dose design, crossover, and then dose titration. And so parallel, for those of you who aren't familiar with that, parallel is, you saw in an earlier slide, one of the presenters had dose 1, dose 2, and so that would basically be parallel, probably a parallel design in which you would put a patient on a dose and then keep them there throughout the study. And you might have a medium dose and you might have a high dose. And that probably led to one of the other comments by somebody earlier in the conference today where they said, do you want to be put on the low dose or, and maybe not get an effect, or do you want to be put on the high dose and have lots of toxicity? So their dose ranging and parallel dose ranging has been used pretty uniformly by the industry since the 1980s, and including for pediatrics. It was, of course, primarily for adults, and we didn't have many pediatric studies before 2000, but they've continued to use this parallel design. The problem is that there are ethical considerations because you have to provide a prospect of direct benefit, as been mentioned by Dr. Weiss, was mentioned in this session. And we found that in 1/3 of the programs that used the parallel dose design, that the lowest dose in the parallel design was actually lower than the approved dose. So the prospect of direct benefit may in fact have not been there, and it may have been questionably ethical to do those studies. Dose titration actually makes a lot more sense, particularly for when you have a small patient population. Dose titration got kind of a bad rap. This is probably 30, 40 years ago with the antihypertensives, when doses were overestimated. The actual needed doses were overestimated for using antihypertensives in adults. And that was probably because of a lack of understanding of the disconnect sometimes between pharmacokinetic and pharmacodynamic effects, the need to do dose titration slowly, and the fact that being slow and having probably optimal doses for patients that were considerably different, perhaps in some cases, doesn't fit the paradigm for drug development industry very well. And dose titration is not necessarily very easy to do, and do properly. there was another comment I heard today say starting doses were too high and advancing too quickly. Well, obviously you've got to start with a dose that has, for a pediatric patient, you've got to start with a dose that has a prospective direct benefit, and then titrate from there. But dose titration has probably the greatest prospect of showing us the optimal dose for individual patients if we use it properly. The other thing I wanted to mention was long-term safety. And one of the earlier speakers said 86% of patients experience toxicity, and they may take these drugs for life. Well, we don't have very good guidance in FDA about how to do long-term safety. The guidance for long-term safety in the US, in the FDA, it was written in 1995. And it was written, so that was before we had pediatric studies. And it suggests that we had to do 600 patients or whatever. Obviously, that doesn't make sense for what we're talking about today. And so I was glad to hear the discussions about registries and doing registries in a better way. They probably are our best prospect of getting good long-term information about safety, and not just about on-target safety. But we often talk in pediatrics about secondary target and pediatric developmental safety. And secondary targets. And so actually, I'll put a plug in for a workshop that we're having at the FDA February 27th and 28th on pediatric developmental safety assessment. And so, long-term safety is something that we need to think about and plan for, and hopefully do a better job of predicting in the preclinical space, which is what we're going to talk about, and also in monitoring long-term, perhaps with registries. Thank you very much.
Gil, thank you so much. N of 1, dose ranging options, including titration, long-term safety guidance. Last time when we saw it was 1995, PED studies didn't even exist there. And also secondary targets. All helpful points. Sander, do we have you this time?
Working now?
Yeah, let me introduce you again now in case folks didn't hear me before. Dr. Alexander Vinks, partner at NDA Partners, goes by Sander. We're in business.
Thank you. And my mic is working now. Thank you again, Garrett. I also want to thank the Duke-Margolis Institute for this wonderful opportunity to participate here this afternoon. This is truly informative, but also very inspiring meeting. So I'm Sander Vinks. I am a professor emeritus pediatrics and clinical pharmacology. I used to run a division of clinical pharmacology and a Center of Excellence in Pediatric Pharmacometrics at Cincinnati Children's Hospital Medical Center. So I am going to focus on some of the things that we've heard today on approaches to dose finding, but then specifically in pediatric studies. And I have heard several speakers and panelists say we need improved tools. I think in the pediatric area, we are using quite a, say, armamentarium of quantitative tools. And this all started with, I think 20 plus years ago with the PPRU, the Pediatric Pharmacology Research Unit Network. That was basically NICHD sponsored, an infrastructural grant type where we had a couple of quants, Jeff Barrett at the Philadelphia Children's Hospital, Edmund Caparelli in San Diego, Steve Stern in Utah, and myself. We started early on with what is now with our now common tools like population pharmacokinetics, physiologically based pharmacokinetic modeling, PK/PD modeling, and try to integrate that. And I think, as Raj said, just the integration and then using that, all that information to not only design very informative trials, where, for instance, if we have very limited patient numbers and we don't want to have a very burdensome trial, we could use optimal sampling theory to come up with very sparse sampling. We can use now these nano devices, these VAMs and dried blood spots, to basically also measure some biomarkers, including drug concentrations, and then build these models that would include the effect of growth and maturation and use them for exposure matching. And we at the team in Cincinnati has been very successful doing this for various rare disease trial designs, but also for what we call model-informed precision dosing, where we do this actually in real time to basically optimize our patients. We've done Pompe disease, we have Prader-Willi designs, Friedreich ataxia, but I think the most successful was in our vascular anomaly program with Denise Adams, where we basically worked out the pharmacokinetics and pharmacodynamics and the developmental pharmacokinetics of sirolimus, one of the mTOR inhibitors, and turned out to be the first effective drug in vascular anomaly that is now also being tested in Sturz-Weber and neurofibromatosis. But we were able to come up with a model-informed platform that could go from trial design to pediatric precision dosing. So it would address some of the limited number issues that we heard today. In terms of heterogeneity and variability, especially the PK variability, can be controlled or much more handled much better that way. Heterogeneity is typically something that more and more is now being analyzed using natural disease history progression, not only data collection, but actually the modeling, because the modeling gives you a mechanistic or a conceptual framework that would basically maximize information and integrate that into these informative models and platforms. And one example that I know CPAT has been working on and has been published in Clinical Pharmacology and Therapeutics and CPT Pharmacometric Systems Pharmacology, where they basically came up with a disease progression model for Duchenne's muscular dystrophy. And I know there is also a clinical group, it's called CTAP. It's the Collective Trajectory Analysis Project, which is, and their mantra is collaborative learning from patient data in rare diseases. And these disease progression trajectories that are basically predicted by the model can then be used even at the individual patient level. So you can then compare placebo or natural history progression versus drug effects, which is a very effective, informative way of bringing together all the relevant data in a clinical trial. For instance, if a primary outcome parameter would have failed, you can still go back and based on, for instance, mutation type, see whether or not there is any benefit for that specific cohort of patients. There have also been a series of very interesting publications recently in clinical pharmacology and therapeutics where they used item response theory modeling versus this natural disease progression and basically looking at trajectories where basically each say element of the rating scale is modeled as a separate trajectory as opposed to a composite endpoint. You can then tease the information apart and basically get a much more granular picture of what potentially could be a drug effect or a signal that is worthwhile pursuing. So those are, in my experience, a couple of things that we have learned from. We are clearly a very strongly biased group in terms of that we think modeling and simulation is a must for every pediatric program, including for every rare disease program. Earlier on, one of the speakers mentioned sickle cell anemia. We have been very successful in using modeling and simulation in sickle cell anemia. And one of my T32 fellows is now rolling out this whole concept, not in the US, but in sub-Saharan Africa, in a truly or resource area where this is going to work. So I'll stop here, but again, very inspiring, and I look forward to our discussion in this session.
Yeah, Sander, it is really inspiring to hear this amount of examples and level of detail. I wonder if I can ask, as I'm summarizing a few of the points that Sander just brought up, if I could ask the rest of the panel to come back on camera we've got about 20 minutes for Q&A and discussion, but it was remarkable to hear the numerous examples just from Sander alone of the improved tools in use for population pharmacokinetics, model-informed precision dosage design, optimal sampling for use in exposure mapping. It's really quite remarkable. Well, let me open up with this one. I want to make sure that we, have a bit more additional specificity on how these dose finding efforts can be tailored to pediatric disease populations. Any additional specific examples of this particular kind of set of use cases related to pediatric rare diseases and finding the right doses? Rajing.
Thank you. Maybe I can start first. So a lot of programs we see at FDA, the sponsor is actually using biomarkers as the primary or even secondary endpoints for those funding studies. So there are many advantages of using biomarkers. For example, the biomarkers is sitting in the physiology pathway of the disease and directly linked to the treatment effect. So its response is usually sooner than the clinical outcome. So from ethical perspective, if the dose is suboptimal dose or is not effective dose, then you can identify it sooner than only looking at a clinical outcome. And also, biomarker has less noise compared to the clinical outcome. So you usually will see the difference using a smaller number of patients compared to you only use the clinical outcome to looking at the treatment effects or finding the right dose for later phase. So yeah, and also the understanding of the dose response or exposure response is really, really important. As the other panelists mentioned earlier, because of the limited number of patients, you cannot repeat it study if it's not ideal or if it's failed. So as much as data you can collect, for example, PK, PD, even in pediatric patients, younger age, if you can have some sparse PK data, that's going to be really helpful to determine the optimal dose or the best for the dose finding outcome, study the outcome for the later phase. Yeah, that's from me. Thank you.
Thomas, let's go to you next. And as you're coming off mute, I can tell you just got a specific question from the audience related to the concept of modeling and teasing out specific parts of endpoints instead of using composite endpoints. Either there or wherever you also wanted to jump in. And Bryce, I'll go to you next.
Yeah, maybe just a quick follow-up on the biomarker topic and specifically a wet biomarker that I very much agree with Dr. Lee that may be a bit more sensitive in discerning change. I think it's important for the sponsors that are participating today to appreciate that the qualification of that biomarker might have been evaluated previously in an adult population. We should not assume that the relative rate of change with disease progression is going to be the same in children. And drilling down even a bit further, we should not assume that the relative rate of change over time with disease progression will be the same in an adolescent versus a child, an infant, or a neonate. So we may need to think about very logical stratification in cohort walkback designs and with qualification steps in advance of a study that's deemed to be pivotal. And your next question was?
You had a direct call out from the audience on concept of modeling and teasing out specific parts of endpoints instead of using composite endpoints.
Yeah, composite endpoints certainly make sense in certain circumstances, but not all. I think for traditional pediatric studies along the lines of that we've been discussing for the most part. Our focus has been on small molecule and maybe first generation biologic based therapies where we have the ability to collect PK samples and measure PD. It may be that an extrapolation-based approach may be adequate in your particular circumstance to support labeling and an indication. So unfortunately, the answer is probably a bit obtuse, as it depends. But there may be circumstances depending upon your drug where a traditional test for efficacy, as you might have contemplated it previously, might not be necessary. And an extrapolation-based approach may secure indication approval.
Bryce, can we go to you next?
Yeah, thank you. And what I'd like to do is sort of pivot from in recognition of the important role that biomarkers play to dose finding there, to the clinical outcome assessments and their role for looking at, because importantly is, you might be able on a biomarker to find the optimal dose there, but if the patient's unable to tolerate the treatment, then it's not going to be an effective dose overall there. And so I want to briefly reflect on some of the work that we've been doing in the pediatric oncology space around assessing adverse events, which is used in oncology trials, in all trials overall. As you know, all AEs are required to be collected, and adverse event includes biomarkers, clinicians' reports, as well as symptom assessments overall. And currently, the standard is that, when it comes to symptom assessment, oncology trials and in other trials there, the clinicians are the one who are grading and reporting adverse events. As I alluded to in previous of my comments, there has been comparison of what the clinicians believe the child's experiencing versus what the parents believe the child's experiencing versus what the child's actually experiencing themselves, her or himself. And consistently, what we find is at best, the clinician has only fair, but sometimes poor relationship to what to the child's actually experiencing. No surprise, the parents have a better reflection of what their child's experiencing. And so recognizing that clinicians typically downgrade and underreport a number of symptom AEs compared to what the child self-reports there is we led initiative funded by the National Cancer Institute to develop a pediatric self-report version of their common terminology criteria for adverse events, or what we call the Pediatric Pro CTCAE. And so this system both allows the children as young as 7 years of age to self-report, but also has a parent observer version as well for parents of younger children to report symptom AEs, and with the goal that this can help improve the validity of adverse event reporting. Pro-CTCA has been sort of supported both by the FDA through their patient voice, by the NCI, and in the Children's Oncology Group overall. So we hope this will continue to be a larger trend to incorporate the patient perspective, or as I said, when the child's too young, the parent perspective. And in addition to having these measures available to the public they're now being embedded in both early phase trials as well as late phase trials. Thank you.
Very helpful. Are there other folks have thoughts on this question of what can be done to collect and report patient outcomes, whether pre-market or post-market, when it comes to dose finding, or did you want to go another route?
Could I just make a quick comment to follow up on Bryce's comments?
Please, yeah.
and related to adverse drug effects? Because extrapolation was brought up here, and now ICH 11A has allowed for extrapolation of safety. You have to do that very cautiously. We've published a couple articles now where we show that the incidence of adverse effects is very different in pediatric patients than it is in adults. And certainly from a developmental standpoint, and that's one of the reasons we're having this developmental workshop, we would expect developmental toxicities to occur that would not be able to be predicted from adult experience. And so I just wanted to kind of make that comment since we're talking about extrapolation.
Comments from others? Thanks, Gil. If not, how about another one of our questions that we had brought up before, and then I'll also look at some of the audience activity here. Many rare diseases are presenting at a very early age. Also talked about this range and outcomes as folks age. With the potential for newborn screening being added as a therapy is available, are there things that we're learning there that can inform dose finding and optimization? Are you chiming in, Bryce or Thomas? Yeah, start with you, Bryce, and then Thomas.
Yeah, go ahead, Bryce.
Oh, I didn't. I'm sorry. My hand was supposed to be lowered. That was from the previous session. I apologize. I'll let Thomas talk. Sorry.
Yeah. So this is a I think a super exciting time in the genetic medicine ecosystem with the advent of and rapid expansion of use of rapid whole genome sequencing, and currently by my last count. I believe this new technology was reimbursed in 13 states as long as a risk factor exists. My expectation for this technology with a incredible diagnostic yield will be useful for more rapid patient identification. For potential clinical trials and certainly early identification of disease for clinical management. So I think this is going to be a huge tailwind for the field of genetic medicine. So I think this will maybe be a potential tool to help identify patients and where patients with rare disease A, B, or C are primarily treated from a sponsor's perspective, but less relevant in the actual determination of optimal dose.
So if I could comment on dose finding down to birth, basically. I did want to bring up the canakinumab example. That was in periodic fever syndrome for a rare disease. And so this was published in CPT maybe 3 years ago. Our experience with canakinumab, in which I think we had 3 patients under 2 years of age and only 1 patient under 1 year of age. And with modeling and simulation and with our understanding of the risk benefit, we actually were able to approve canakinumab right down to birth. And so this is a win for modeling and simulation in terms of dosing. Obviously, the more we can understand ontogeny of metabolizing enzymes, now we're starting to work on drug absorption and understanding drug absorption better, and even below 1 year of age. And so, modeling and simulation is going to be it has already been a big step forward for pediatric drug development, and it will be for rare diseases, I'm sure.
Sander?
And I just want to echo that and basically give another example. So I mentioned this mTOR inhibitor for vascular anomaly where we started at age 4 or 5, but then worked our way back and were able to basically elucidate the ontogeny of drug metabolism of sirolimus and are now very comfortable of dosing newborns on 2 days of age. Actually, Cincinnati Children's has a service that, we get requests now through Denise Adams from all over the world. So we help physicians and parents in Singapore and Japan when they actually don't get the dose right for especially these very young patients. And I think this is a beautiful example where modeling and simulation can truly visualize where you are, where you want to go, and how you get there. And again, you can confirm what the model is predicting because typically these institutions have analytical capabilities. They have assays for sirolimus because it's also widely used in transplantation. And as we know, in transplant patients, there is a strong incentive to have therapeutic drug monitoring be part of the whole therapeutic strategy. So that is available. And I think we can learn from those infrastructure, say, resources that basically would help do some of the biomarker / drug concentration measurements as part of those optimizations. Again, we've been very successful in doing model-informed, exposure-controlled or concentration-controlled clinical studies. We're currently working with a sponsor on one for Prader-Willi syndrome, where we have developed a PK/PD model and where we target exposures based on receptor occupancy of their drug. And so, and that is a very sophisticated way of doing a dose escalation study, if you will, but it's model informed and model controlled. So you have a much better understanding of what is going on and you get feedback. And once you get feedback, you can use Bayesian estimation to basically go from where you were to where you want to go. I think those are some examples that are very useful to keep in mind when developing a program for a orphan or a rare disease pediatric program.
Yeah, once again, inspiring, hopeful, and just the level of specificity to multiple examples is really encouraging. We've just got a few minutes left. Can I give kind of one final pass for the most basic of them all? How do you develop these dosage regimens when data are not available? We've gotten lots of good examples so far. Are there others that we want to make sure that we put out on the table now before closing up for the day, and then hopefully lots of great activities to follow? Thomas.
Yeah, maybe just one thought for the audience to take home as they think about their own initiatives. I noticed one of the questions in the chat referenced within patient population heterogeneity, and this is adds a different level of complexity for us that are trying to determine whether or not we have a treatment effect or not. This is a topic that we've been giving considerable thought to as of late, and how could we be thinking about design of early or first in children trials where we think about endotyping the patient population and trying to strip out heterogeneity by focusing on patient subtype and exposure. And in some circumstances, we've learned that dosing will be consistent across disease subtype. In other circumstances, that's not the case. So really thinking through, particularly with new tools that we have that we all can avail ourselves of in AI interfaces with very large data sets, might we be able to subset these populations a priori and understand whether or not dose response is in fact the same or not in each patient subset. And that's if you're concerned about heterogeneity on the front end, maybe one strategy to think about to try to overcome.
Appreciate that. Anyone else from the panel, either on that final question or just closing remarks?
Well, I would just perhaps comment that we still follow Lou Shiner, learn and confirm, and so we although we're very strong supporters of modeling and simulation, and they've done wonders for our pediatric population, we still are cautious regulators, and so we will probably ask for some confirmation at some point about the doses that we model.
Sure.
Sorry, maybe I can be the next. So I echo other FDA panelists mentioned earlier that as FDA, we always encourage the sponsor to get engaged with FDA at the earlier phase. So because for pediatric rare disease, as everybody already mentioned, there's so many uncertainties like biomarker, heterogeneity, and it's good to have an early discussion, even for example, pediatric extrapolation, whether this case is feasible for pediatric extrapolation, we can all have the discussion at early phase, so we can help the sponsor to plan ahead of time for the best regulatory outcome for the program.
Can never be said enough. Thank you, Rojang. Bryce.
Yeah, no, I just wanted to follow up that comment there and just talk about we've had some very rich and thoughtful interactions and collaborations with the FDA. We're funded in 2 FDA projects where we had the opportunity to collaborate with them to help integrate clinical measures in pediatric rare populations there. One is we're working with the FDA to develop measures of pain in neonates and young children, capturing both the clinician perspective as well as the parent perspective in these things overall. I also want to highlight our collaboration with the FDA on development of measures of communication functioning in children with neurodevelopmental disorders. This was work originally funded by the Foundation for Angelman Syndrome Therapeutics, but represents how we can integrate these measures in rare pediatric populations, capturing unique things there that we know is important to the parent's perspective. So again, I think there's unique opportunities, and the FDA has been more than helpful to in making sure that we have regulatory grades measured for use in these rare trials.
Great examples. Sander, last, if you got anything to close us out, and then I'll hand back to my colleague, Victoria Jim, to close out the day.
I have a couple of things I just want to highlight. One is the learn, confirm, and for me, the apply piece. And so it's learn, confirm, apply, and then feedback to, and that comes back to what you were saying, Gil. Yes, make sure that it truly is safe and effective, but also for the long term. In terms of, and we've heard this several times today, collecting data, I think we have the infrastructure, IT systems that could do this. Bryce, you mentioned pain management. When I was at Children's, we developed a pain management system for morphine in our neonatal NICU, our neonatal intensive care unit. And we built this into our electronic health record system, where we showed at the same time the pain scores and the predicted and measured morphine concentrations as if you were seeing your ECG readout or your heart rate on the monitor. Yeah. And I think that is something we would want to build for rare disease, well, for any disease, but definitely for rare disease programs, where you can, in real time, collect this type of information, but also feed it back to, for instance, the PI or the research team, or for the people that do the, the adaptive design, so you can truly follow this. Because we're looking at doses, but patients change. I mean, for instance, from the sirolimus trial, we learned that if they get infections, their cytochrome P450 system is downregulated and the drug levels go up, at least by 100%. Well, we learned this, so we say, well, half the dose during the infection period, and you've done. But those type of things you would not see if you don't collect that data and visualize it in a meaningful way. And I think we have the technology, we have the platforms, we now have to have smart people that basically put this together and see this as an important next step of building these very informative clinical trial systems that basically support this type of drug development. I'll stop here, but thank you.
Yeah, lots accomplished, but lots still to do. Thank you all very much for participating today, and frankly, for the decades of work from each of you participating in a panel like today. I hope to catch you again before too long, but for now, I'm going to turn it back to my colleague, Victoria Jim, to close out the day. Thanks again.
Great. Thanks so much, Garrett. I'll now take a moment to close out on what has really been such a fantastic day of discussions, a lot of really rich information shared throughout. So many thanks to our panelists, moderators, and attendees as well for such an engaging event. I know there were a lot of questions submitted. We weren't able to get to many of them, but we'll certainly take those back and really appreciate having those questions as we continue to inform this work. I want to thank each of you for participating, for your thoughtful contributions, and then we're just very fortunate to have such a distinguished group of experts with us today, and we really value the perspectives you've shared. So just some quick recap on some of the key themes and topics we heard throughout the day. We heard about the truly like critical importance of reducing patient and caregiver burdens through innovative trial designs and technologies, optimizing dosing based on patients' risk tolerance and not relying on maximum tolerated dose. We also heard about the importance of utilizing natural history in registries, while also acknowledging some real-world data is not collected with studies in mind, and some balance and caution needed there. Utilizing standardized data approaches, including federated data models to support relevant research, as well as strategies to address doses for various pediatric development phases and outcomes considerations. So a lot covered today, a lot of nuance, a lot of caution and opportunities shared throughout. So really appreciate those. So I would like to take a moment also to thank several people for making this event possible. I'd like to thank the FDA staff that played an instrumental role in planning this meeting. Really was truly a joint effort. So many thanks to Robert Chuck, Michael Pakinowski, and Rojang Lee. I would also like to thank the Duke-Margolis planning team, Val Parker and Mia Williams. I'd like to thank the Duke-Margolis staff who assisted in everything that took leading up to today's event, as well as supporting throughout today's event, including Maddie Cordell, Hannah Vitello, and Luke DeRocher. And then as a reminder to everyone, additional meeting materials will be posted on the Duke-Margolis website in the coming days, including the recording of this event. So thank you again, and hope everyone has a great rest of your afternoon.